morning. Welcome, thank you. Everyone can hear me okay? Okay, great. I'm Michael Rosen. I have the privilege of directing the Stanford Medicine Children's Health Center for IBD and Celiac disease, and I'm excited to welcome you today to our second annual research symposium. I want to start, as we always do, with our land acknowledgement, which is that we recognize that Stanford sits on the ancestral land of the Muwekma Ohlone tribe. This land was and continues to be of great importance to the Ohlone people. We are, as always, incredibly grateful for the generosity of an anonymous donor for supporting the vision of a world-class clinical and research center for pediatric IBD and celiac disease here at Stanford, and for making it possible for us to come together today as a scientific community, as a community committed to advancing the understanding and treatment of these disorders in children. And the mission of this center is to integrate leading-edge clinical care and research to elevate the lives of all children confronting IBD and celiac disease. Just to orient you all to how we envision the structure of this center, it's really organized at a high level into three major pillars. On the one end, we have care, and research on the other end, and really connecting these two and bridging them together is our patient biorepository. We have transformed clinical care at Stanford with faculty recruitments, assembling an interdisciplinary care team, making investments in technology and advanced training for these disorders. And then within our research pillar, we are also have ongoing and existing new recruits, faculty recruits that we're excited about. And of course, we have a tremendous intramural research grants program across campus, which includes both seed grants and early career training awards. And again, to integrate care and research, we've built a biorepository, and I think when we spoke last year, it was really in its inception. But now, really to the tremendous effort of our clinical research coordinator team, we've grown this biorepository to nearly 100 patients enrolled with hundreds of specimens, including blood, tissue, and stool. So in collaboration with MCHRI, the Maternal Child Health Research Institute, we've awarded 19 grants for a total of $4.9 million. This is really a tremendous opportunity. You'll hear from one of our grant awardees, Michael Howitt, today in his research. We have new clinical leadership I'd like to introduce you to. So since our last symposium, Hillary Jericho is our medical director for the celiac disease program, joined us from University of Chicago in January. And just in August, Jonathan Moses is our medical director for the inflammatory bowel disease program, and he joins us from Rainbow Babies and Children's at Case Western. So welcome, Hillary and John. And then I also want to take a moment to thank the incredible administrative leadership and team that really made today possible in terms of all the planning and organizing. Ashley Dunn, our program director, who really makes everything tick within the center, and Alexandra Lazardo, our executive administrative assistant, who led much of the planning for today. Thank you, Ashley and Alexandra, and thank you to our clinical research coordinator team and other admins for your help today, making today such a great success, I'm sure. I'd like to introduce you to our new website, which is really, again, the effort of our administrative team, in this case led by Jasmine Yuen. So we have our new center website here, and what I want to draw particular attention to is please go to the website, click on People and Membership, and if you haven't already joined and become a member, please complete a short application for membership, and you will then be updated about grant opportunities, events like these and others. We'll seek to update folks about extramural grant opportunities, and this will enable you to be eligible to collaborate for biorepository specimens. So please take a moment, maybe even during a break, to join our center officially as a member. I'd like to acknowledge our External Advisory Board committee members, several of whom have traveled to be with us here today, and some are speaking. I'll let you see them on the slides. Here today with us are Maria Olivia Hemker, Michael Kappelman, and Alessio Fasano. And thrilled to have two of our External Advisory Board members as keynote speakers today, Alessio Fasano from Harvard and Mass General, who'll be talking about celiac disease, and Maria Olivia Hemker from Johns Hopkins, who'll be talking about C. difficile and IBD. So really, in just a few short years, I have been inspired by the members of this remarkable Stanford community who've come together and really come to us and saying, look, we're here, we think we can make a real difference for children with IBD and celiac disease, and we want to work with you. It's been incredibly heartening, and that's why I'm so excited for today, a day when we can come together from all these different disciplines as one community to learn together and ignite new ideas and collaboration. So thank you for being here. And with that, I'd like to turn the podium over to our moderator for the first set of talks, Stefan Regalo from Adult Gastroenterology, who will introduce us to the first speakers. Thank you.
Good morning, everyone. It's a pleasure being here. So I have the pleasure to introduce the first speaker. That's Michael Howard from Stanford. Michael got his PhD from Stanford. He has his Bachelor of Arts in Cell and Molecular Biology from Berkeley, and he did his postdoctoral fellowship at Harvard. His lab is working primarily on the interplay of intestinal microbes, how they're promoting health, how they're promoting disease potentially, and I'm actually very excited to welcome Michael here on the stage as well. So Michael, all yours here.
Let's see. Good morning, everybody. I have this lapel on, so I'm untethered. Well, thank you for the introduction. Appreciate the opportunity to speak today. As Michael said, our lab is sort of moving into the IBD space, and so we're really grateful for the support that this center has provided to us into sort of launching this new avenue of research. And so Stefan mentioned our lab is sort of anchored in a couple key areas. The first is studying the microbiota, and this is a topic that, of course, is familiar to probably many of you in this room. We know that the microbiota is vitally important for our health, aiding us in our nutrition, tissue development, and interest in our lab. How does this affect the development of the immune system? But of course, we also know that the microbiome can contribute to deleterious conditions, of course, notably inflammatory bowel disease. Now, when I started working on the microbiome as a postdoc in Wendy Garrett's lab at Harvard, my sort of thinking sort of started and ended with bacteria, right? To me, microbiome equaled bacteria. But as hopefully I'll try to convince you in the course of today that this is a much more diverse ecosystem with important players that are viral, there's archaeal, and then there's eukaryotic species fungi, and what we're going to be talking about, protists. And so when I began to study this as a postdoc, I was really interested in the epithelium. I wanted to know how was the gut microbiota interacting with the epithelial cells, and what impact did this have on host health and host disease. And so this is sort of the state of the landscape when I began. This is probably something that, again, that is quite familiar to folks in this room, which is that you have the common stem cells here, in the crypt, and that this gives rise to sort of fully differentiated cell types, the absorptive enterocytes, mucus-producing goblet cells, enteroendocrine cells which produce hormones, and then Panna cells. And so this was sort of the landscape when I started. But there was beginning to be inklings of a fifth cell type that we didn't know the function of, and that was the tough cell, right? And the tough cell is so named because of this very prominent sort of tuft you can see actually protruding, this very long microvilli that they protrude into the gut lumen. And basically this cell had been noted because of its very distinct morphological characteristics, but we didn't know anything about what it did. In fact, most people didn't even know that it existed. It's not, however, a new cell type. It was first described in 1955. I recently attended a tough cell meeting, and someone pointed out that maybe it actually predates this to the 1920s, but the paper is in German, and I don't read German, so I don't know. But importantly, tough cells are not sort of an obscure cell type just in maybe an animal model. These are present widely, or at least cells that look like tough cells, across most vertebrates, humans, mice, fish, amphibians, right? They all have cells that are either tough cells or very tough cell-like. But they're rare, right? When I started working on them, the first estimates had just come out suggesting they were about 0.3% of the small intestinal epithelium. So these are one of the rarest cell types in the gut. But it's this last part that actually really kind of interested me and got me excited to actually start studying them, which is the idea that these were taste cells, right? That they had the same molecular machinery that you have in your tongue is present in these cells in the gut. And this was a really intriguing idea to me for a couple of reasons. One, I thought it was incredibly strange and weird and attractive that you had, these taste cells at these mucosal surfaces. The other thing you could imagine is that, obviously we think about taste in terms of food preference, and that may have a benefit in the gut. But there had been also some recent research coming out of the upper respiratory tract in both humans and mice, suggesting that the taste chemosensory system could respond to microbial cues. And when we think about the gut microbiota, this very dense, complex, diverse microbial ecosystem, I was intrigued by the idea that perhaps these taste cells could be tasting microbes in the gut and then communicating that to other cells, right? And so I began to look at this. This was kind of a weird idea. This wasn't my main project as a postdoc, but my advisor, Wendy, supported me in doing that. And so we just did this very simple experiment when she said, can you just even see these weird taste cells in our mice? And so we stained them with a marker called DCLK1. It's not important. It's just a marker for tough cells. And we could see them in the small intestine of our mice. When we counted them, however, they were about 7%, right? So that's a huge discrepancy from that 0.3% that had been reported, suggesting that something weird was happening. Although we were looking in the same region of the gut, the same strain of mice, and with the same marker, we were getting a very, very different result. So then we looked at mice sourced from a different vivarium, right? In this case, ordered from Jackson Laboratories. And we saw way less tough cells, right? They were less than 1%, much more akin to what had been previously reported, suggesting that there was something odd about our mice at Harvard, right? And so, we went through the usual battery of, what could be the environmental differences, the food, et cetera, et cetera, et cetera. But of course, what about the microbiome, right? And so we decided to do something just very crude and a little bit disgusting, which was to take the fecal contents of our bred and house mice, feed it to these Jackson animals, and see what would happen. And lo and behold, we sacrificed them 3 weeks later, and we saw this huge expansion of the tough cells, basically phenocopying what we had seen inherently within our bred and house mice. And so, again, like any good microbiome scientist at the time, I knew it was the bacteria, so we started doing 16S, and we found tons of changes, as you would expect. And so I went and diligently began to test those microbial changes, yet we didn't see anything that was affecting the number of tough cells. And it wasn't until I actually started looking at these tissues by histology that I noticed that all of our bred and house mice had what appeared to me like these little protozoa in them, and these were entirely absent from these Jackson animals. And so if you purify them, you look at them by scanning electron microscopy, you can see that they're rather beautiful looking microbes, these tritrichomonads. We confirmed their identity using molecular techniques as well, and they are something called Tritrichomonas musculus. Again, going to the literature at the time, what did we know about these organisms? Well, again, not much, right? So I was studying sort of an obscure cell with an obscure microbe. They're normal in the mouse microbiome, but they're thought to be benign, to not have any effect on what these mice do and what their immune system behaves like. However, I was able to purify them. I was able to introduce them back into these protist-naive animals, and we basically phenocopied this. So we saw that you saw this big expansion. So they were the organisms driving this tough cell response in our mice. Now, while we didn't know what sort of function they had, they actually proved to be quite useful. And so I'm going to cut to the chase and say that we used these organisms along with intestinal helminths to begin to figure out what was the function of tough cells. And so I'm just summarizing for the sake of time a whole lot of work that we and others have done to figure out the function of tough cells. Basically, what we found is that both intestinal helminths and these tritrichomonas protists stimulate tough cells. The tritrichomonads stimulate them through production of a metabolite called succinate. We still don't know what worms are producing to stimulate tough cells, so that's still an outstanding question. And that tough cells then use the sense of taste to respond to these organisms. They produce a cytokine called IL-25. This primarily communicates with an innate lymphoid cell called ILC2s, and that these sort of elaborate all of the usual type 2 immune effects that you think of: goblet cell hyperplasia, granulocyte infiltration, Th2 responses. All of these are really go through sort of tough cells as the gatekeeper, right? So tough cells are actually the innate immune sensing cell that drives type 2 immunity and these mucosal surfaces, right? And so this was a really interesting question. It's something that we are continuing to work on. But essentially, all the work that we and sort of others in the field have been doing was looking at what was sort of the host side consequences of these tritrichomonads. But what we realized is that we didn't know anything about the protists themselves, right? Essentially, people were publishing studies, and there would be some slight differences. We weren't aware, were these the same species? Were they different strains? What, if anything, was the impact of different microbiotas or diets on these processes? We really knew absolutely nothing about them. We had no genomes. We couldn't grow them. Essentially, all of the usual sort of molecular and wet lab microbiology tools were absolutely not available for us. And so what we were interested in was, could there be unknown diversity? And importantly, as we're foreshadowing with these lines, could they be doing other things other than just sort of little swimming bags of succinate, right? And so to do this, I just want to first point out some of the folks that actually did the work. This was led by a fantastic postdoc, Eli Garrett, and he's had tremendous help from an LSRP1 in my lab, Claire Meckler. So these are the folks that actually did it. I'm just taking credit for their work. And so one of the things that we wanted to do is when I moved from Harvard to Stanford, everyone said, look, you're going to have enormous microbiome challenges. So your phenotypes are all going to go sideways. To a certain extent, that has been true. But the other thing that we hope to do, right, is to sort of make lemonade out of lemons, right? And the idea was that maybe if we're moving to a different environment, we can sort of check for these potential, extra trichomonads that you might have on the West Coast that we were missing in Boston. So we went through and we got stool samples from other people's mice. I even convinced the veterinary folks to let me check the sentinel mice. We saw nothing. So for those of you who are working with mice, you'll be pleased to know our facility is quite clean for these tritrichomonads. It turned out what we needed to actually do is look inward. If we actually studied our own colony, the colony that I had brought over from Boston, we actually had extra tritrichomonads that we didn't know about. And so one of the things that Eli had noticed is that there was multiple sizes of tritrichomonads. And so what you can see here is just a flow cytometry. And this is just showing that there are different sizes. And so we're representing one is blue and one is pink. We sorted these out by size. We introduced them into naive mice to see if we could propagate them. And lo and behold, the big tritrichomonads stayed. We only had the big ones. The little ones stayed little. So these weren't just sort of subtypes of the same species. And we went on to actually characterize them. We generated the first genome sequences along with Ami Bhatt's lab. And so you can just see one we named is Tritrichomonas musculus. The other one Eli named Tritrichomonas casperi. They have hugely different genomes, right? So you can just look at the megabase sizes. They're about 160 versus 64. So these are clearly different species. They're different sizes. You can kind of see them by electron microscopy. They look morphologically very similar. But as I'll show you, they're actually quite distinct properties. You can just see them in terms of the phylogenetics. They are still quite closely related, though. Okay, so we found this new tritrichomonad. The first thing, of course, we wanted to do is say, what happens to tough cells, right? This is the whole reason why we got interested in these protists, is because of their effect on tough cells. And for the very first time, we actually found a tritrichomonad that did not drive tough cell hyperplasia. That expansion of tough cells that I showed you did not happen with T. casperi, nor did it drive a Th2 response, which I'm showing you on the right. So there wasn't the epithelial response, there wasn't the canonical type 2 immune response associated with this. And this is where I actually started getting quite interested in this protist. So he said, okay, if it's not driving type 2 immunity, does this mean it's immunologically inert? Is it just sort of failing to do that thing that other protist can do? And the answer is no, it's not. It actually does drive an immune response in the small intestine. It's driving a Th1 and a Th17 response. And so you can see that it is basically occupying the same niche. It is affecting the mucosal immune response, but in a very, very different way than T. musculus. And so the question is, why is this happening, right? And so to do that, we started looking into the tritrichomonas metabolism, right? And this is an area that Eli is fascinated with. I'm fascinated with it only because I think it affects the immune system, but he's taught me a lot. And one of the things that we learned in putting together, and this is a very simplified schematic, metabolism is an incredibly thorny subject, but they need to as part of their metabolism of carbohydrates, they need to regenerate NAD+. And so in order to do that, they generate tons of succinate, right? So they take fumarate reductase, they take fumarate, and they turn it into succinate, and they excrete that succinate. So about 50% of the carbon they're intaking is being pushed out in the form of succinate, hence stimulating the tough cells. And so, again, when you measure by mass spec, if you look in a mouse that's colonized with T-mu, there's tons of succinate in the gut, basically nothing in the T. casperi, again, showing that the tough cells were reflecting, being very good succinate sensors. So when we looked at the genomes, we put together their metabolic pathways. So in red, you can see the genes that are expressed by T. casperi. In blue, it's T. musculus. T. casperi actually encoded for both the succinate production and an alternative redox pathway that generated lactate. And so we thought, there's our answer, right? It's making lactate instead of succinate. And so when we looked, the answer is it's not, actually. And that actually is in keeping with when we looked at actually gene expression. They're expressing far more fumarate reductase. That gene is expressed far higher than, say, the lactate dehydrogenase. So this all kind of makes some sense. When we actually cracked open these protists to see what was inside them, we actually saw that they were both making the same amount of succinate. And so what we believe is happening, and this is still something that we don't entirely understand, is that they're actually making succinate, but that they're converting it to something else, and that is actually being excreted out, right? And so we don't actually, unfortunately, yet know what the identity of that is. Right, so here's kind of where we're at, right? So we discovered a new organism. We know that it has a different metabolism, which can affect its ability or inability to drive tough cell-mediated immune responses. The other big question is what, if any, what is the interaction with the microbiome and with diet, right? How does this impact the situation? And so I'll show you just a quick set of experiments. We turned from using a complex mouse chow diet to defined diets, right? And so in this case, we had sort of two carbohydrate sources. One was fermentable, it's called inulin. The other was not fermentable, called cellulose, just sort of a packing agent, right? And so when we look at T. musculus numbers, and so you have complex is just normal mouse chow. The defined CI, the C is cellulose, I is inulin, and then we removed all fiber, and then we only had cellulose, or we only had inulin as the fermentable fibers. And what you can see is basically the T. musculus, it needs to have fermentable fibers to survive. It dies quite quickly when you remove fiber. To our surprise, T. casperi doesn't care. You can totally pull out fiber. You can give it unfermentable fibers. It doesn't change its colonization whatsoever, suggesting that it is not relying on the diet for its food. So what could it be doing? And so we thought about, well, what are the other sources that are present? There's tons of host carbohydrates. And so we went, and one of our first sort of approaches to understand what it could be is, instead of trying to figure out what's going on with the protist, we thought about, well, okay, if these aren't going into an intact microbial ecosystem, can we look at what they're possibly displacing to get clues about what nutritional niche they're occupying, right? Because again, we knew hardly anything about these protists. And so when we did 16S on mice colonized with T. casperi, this is a left subplot. So basically, what you can see in green is what is relatively enriched, and what you can see in red is what is relatively depleted. And you can see two phyla of bacteria, Bacteriales and Akkermansia mucinophilia. as the name muciniphily would suggest, this organism likes to eat mucus. When we confirmed these, specifically looking at Akkermansia, we could see that T. casperi essentially eliminates it. So T. casperi is competing with mucus eaters, suggesting that perhaps this organism is actually surviving on mucus and why it doesn't need dietary fiber. And so when we think about mucus, of course, we think about the mucins, which are proteins, but these are actually, importantly, 80% carbohydrates, right? So this is a really rich source of polysaccharides that is potentially available to microbes. The thing is, these are very complex structures, and you need to have the right enzymes to actually liberate these sugars. And so we don't know much about this, again, in the protists, but we do in bacteria. And so we went and we looked through some of the different glycoside hydrolases that potentially these protists could be expressing, and we used some predictions to understand whether these are host-derived polysaccharides or dietary-derived polysaccharides. And what we could see is that T. casperi is expressing all of the major glycoside hydrolases necessary to liberate some of these sugars off of the mucins. So that looked fantastic. The downside, however, is that we also looked at T-mu, which we thought was this fiber eater from the diet. And while the genes were lower, it was still expressing some of these genes, right? So why is that, right? If this is, why would they express these putative mucus-eating genes? And so to really start looking at this, Eli made a breakthrough that I would guess maybe refer to as sort of the most exciting piece of boring data that he's shown me, which is essentially just a growth curve, right? And very few people could appreciate how awesome this is, because I had worked on this for a couple of years. Eli had worked on this for a couple of years. And to our knowledge, still no one other than our lab has been able to grow these things. And so just this very simple thing has been incredible. And it's allowed us to ask some very specific mechanistic questions, such as like, can they actually eat mucins, right? And so Eli took this advance, and he can grow them using maltose as sort of the primary carbohydrate. They love that. But he also substituted out maltose for mucus, right, as their sole carbohydrate in vitro, and they grow great. They love it, right, which would be in keeping with the fact that they express some of these glycoside hydrolases associated with liberating these sugars. And so then we wanted to know, okay, so if they express the right genes, they can grow on mucus in vitro. Why are they having these issues in vivo? And so what we went and if you look at them, right, so if you look at normal complex chow, in green up there, that's the protists, right? So they're sort of spread out throughout the gut, right? They like to eat the food, so that's where it's present. If you put them onto cellulose, which is, again, a non-fermentable carbohydrate, there's very, very few of them. You see them drop out, oftentimes below detectable levels, but there are actually a few of them remaining, and you can see them associated with the mucus layer. However, there's very few of them. And if you then put those mice back onto a complex chow, you'll see these protists will sort of bloom out from there. And so what we wondered is, what kind of the key differences could be the fact that obviously in the test tube we didn't have the microbiota, right? And so we decided, okay, could we go and kill the bacteria, and could that potentially help T. musculus in these fiber-deprived conditions? And so essentially we gave mice cellulose again. We fed them antibiotics, Vanc, Neo, and AMP, so this sort of broad spectrum treatment, and we saw this total rescue of T-mu, indicating that if we could get rid of the bacteria, this protist was perfectly capable of surviving without fermentable microbes. And so then to kind of begin to tease apart a little bit more who it was, we then broke those apart, and we saw that basically it was ampicillin. That was what was rescuing T. musculus from this. And so, again, we did a bunch of analysis, and I'm just going to distill it down into what we think is probably the most important element of it, which is that only when we treated with ampicillin did we get rid of the mucus eaters, right? So in all the other antibiotic conditions, obviously we did a lot of damage to the microbiota, but there was always some other microbe that was really good at eating mucus remained, right, except for an AMP. And so basically, in summary, and I'll kind of get to a little bit more after this, what we found is there are different microbes, right, present, different protists present, excuse me, within the mouse microbiome, that these things rely on different carbohydrate sources. Sometimes, in the case of T. musculus, it's dietary fiber as a preferred source, and I didn't show this to you, but they actually can outcompete fiber-utilizing bacteria under those conditions, right? Whereas other organisms like T. casperi are principally relying on mucus. They're more mucus specialists, and in turn, they compete against other mucus-consuming microbes like Akkermansia and mucinophilia. That T. musculus drives a Th2 response where T. casperi, for instance, does not because of these metabolic differences. Other data that I did not show you is actually there are some conserved features between them that allow them both to drive a Th1 and Th17 response in the colon. So there's some more there that I didn't get to. But that by modulating the diet, you can basically alter their abundance. So that if you remove fiber, that we see T. musculus is basically outcompeted by microbes and it goes away, versus T. casperi is unaffected. We added back specific fibers, so we sort of defined the fibers that T. musculus can and cannot eat. And then if you add inulin back, for instance, T. musculus can actually not eat inulin, it turns out. What it does is we believe it's distracting some of the Bacteroidetes. They prefer to eat inulin. That basically, they then vacate the mucus niche and allows T. musculus to then occupy that. However, one of the things that we noticed is that when it adopts that different metabolism and that different lifestyle, you have a reduced type 2 immune response. In other words, you can modulate the immune response by modulating the diet as well. And so this kind of leads us very briefly to sort of how do we envision this potentially impacting IBD? And the first thing is, great, nice studies on mice. Do humans even have these, right? And so that was a pretty big open question. There had been some reports of parabacillid protists, which include these tritrichomonads, such as Pentatrichomonas hominis and Dientamoeba fragilis. But we really didn't know hardly anything about this in people. And so in collaboration with the Sonnenberg lab, we reanalyzed metagenomics from about 1,800 different patients across 29 different populations. Importantly, these populations span the globe in a number of different lifestyles, from hunter-gatherers, transitional, and to industrialized populations. And we saw a couple things that we think are really cool about this. One, we actually got, I think, the first large-scale sense of what is the prevalence of these. And unsurprisingly, in industrialized populations, the prevalence is quite low. So we were seeing below 4%, and the diversity is also quite low. D. fragilis is sort of the dominant species. However, if you start looking in non-industrialized populations, you see the prevalence goes way, way, way up, right, 30%. And to our delight, we also saw that there's a ton of different species that we had never encountered before. And so just to kind of to help you understand this. So when I, for instance, show, you can see there's Trichomonas vaginalis, we're not suggesting that these people have T. vaginalis in their guts. What we're suggesting is that there are organisms there that are new, and that the closest relative that we can see that has genomic sequence is T. vaginalis. The same thing when you look at T. casperi or T. musculus. We're not suggesting that these rodent protists are zoonotic, just that these are new species in people, and these are the closest things we have. We've also assembled some ITS sequences from this metagenomic data, and we've actually made some new calls of some definitely some new species, potentially some new classes. And so the important point here is these are in people, they're relatively prevalent in non-industrialized populations, and they're quite diverse, and there's a lot that we don't know about what these organisms are doing. Importantly, we have a framework, a phylogenetic framework that we developed from both animal and human sources, where we've been able to either use sort of genetics or metabolomics to begin to understand the immunomodulatory products, so whether they're producing lactate, which can modulate the immune system, or succinate, which can modulate the immune system. And so that way we can just use phylogenetic relationships to make some reasonable predictions about what these things might be doing in terms of at least two important metabolites. Obviously, as I highlighted with T. casperi and T. musculus, even close relatives can sort of buck that trend, so it's imperfect, but we have some sense of how it works. And then lastly, tough cells, right? And so from work that Ken Lau's lab has done at Vanderbilt, they went and they looked at tough cells in patients, and they've also looked, with Crohn's disease in adult patients, and they've also looked in mouse models of inflammation. They found that the tough cells were decreased in these patients. They found that in mouse models, if you feed them succinate, you can ameliorate some of the inflammation. And so suggesting that perhaps tough cells are an important lever, which we can pull on the mucosal immune system, and that potentially these protists are useful ways that we can actually modulate that. And so we're very interested in sort of understanding how are tough cells affected in pediatric Crohn's, what are the tough cell sort of receptors in biology, both in the small intestine and the colon. These are actually very, very different in regards to tough cells. And so how might these be potentially useful, and how might we also understand how these protists are interacting with them to either understand the protists themselves or potential factors they're making beyond succinate to modulate this. So again, I just want to thank everyone who's involved, particularly Eli and Claire. We've had some fantastic collaborators. Ami Bott's group has been absolutely essential to this, as has Justin Sonnenberg and his group. So thank you very much.
Thank you so much,
Sure.
Michael. Are there any questions from the audience? I guess Michael.
Is it on? Ooh, okay. Wonderful talk. Thank you, Michael. So in inflammatory bowel disease, particularly in ulcerative colitis, it's been well described that there seems to be a augmented type 2 immune response in some patients, and I think it likely confers better outcomes in those patients. And then in both Crohn's disease and ulcerative colitis, in different scenarios, we can see eosinophils, and I think it's debated sort of as is always with eosinophils, what their role are, and maybe there are different subtypes of eosinophils doing different things. But then nonetheless, do you think there is a possibility that some of this can be regulated by protists?
Yeah. So yeah, it's an interesting question for two reasons that we think that's actually particularly interesting. So one is that this succinate response is specific to the distal small intestine. Colonic tough cells are actually not sensitive to succinate. However, so in other words, succinate isn't going to be a useful lever for the colon. However, we can see through yet an unknown factor that we don't know that these protists can actually stimulate type 2 immunity in the colon independent of succinate, but dependent on tough cells. So we do think these are going to be a useful sort of avenue of inquiry there. We have, so Claire has actually started a project looking at the way that these protists may, through tough cells, influence colitis, and she is seeing potentially some benefit, though these are very early days. So yeah, so we do think that there's some potential there, and we think that the protists are going to be the way to get at it, as opposed to succinate. But yes, so we think that's potentially interesting, yeah.
Michael.
Yeah, fantastic talk, really exciting. We have a mouse model of enteroid epithelial death, and when we grow the organoids, we could see things swimming around in there. And with the testing, it was, I think, we thought it was T. muris and spironucleus.
Yeah.
And so I was just wondering if, I guess, if you have spironucleus here, if there's any role of like other protists kind of in the upper GI tract in terms of this kind of biology, or.
Yeah, so, you could show me a video and I could probably tell you what it was. But yeah, so we don't have spironucleus muris anymore. This is something that we did have, Harvard had them all. And so we did have it, and I have looked at Spiro on its own. It does not drive this response. The fact that you're getting these organoids out means that you're probably grabbing them out out of the crypts, right? And so spironucleus muris, unlike these protists, which tend to sort of hang out above the mucus layer that don't penetrate down into the crypts, you're very likely, because I've noticed the same thing, it's very likely spironucleus muris is what you're seeing swimming around in there. Sorry.
It doesn't drive tough cells.
it does not drive a tough cell response. So it is interesting to think about what it could be doing, yeah.
Okay, one last question. I think you were first.
I was wondering if you could speculate a little about the relation between structure and function in these most unusual looking epithelial cells.
Yeah, so yeah, it's a great question. So there's these things are structurally very cool looking, not only because of the tufts, but they actually have these axiostyles, these spikes they send out to the sides that actually jam into the cells next to them. We have no idea what they're doing. They almost go to the nucleus but without penetrating, so that's an interesting thing. They also have these little tails that come inward that almost look like little dendrites. So there's some interest in whether they could be in any way connecting, or whether these could be probably not forming synaptic connections, but potentially influencing the nervous system. Regarding what I showed you, which is the villi and the cytoskeleton, which I didn't point out, they also look like mechanosensory cells as well. And so there's some thought when you look about where these are in the body, not just the gut, but some of the, like a lot of ducts, have them. And so that we think that there's probably also some mechanosensory properties as well. And we can see that these things are modulating other non-immune factors, potentially like motility, contraction. They may make acetylcholine, for instance, fluid secretion. So we do think that there are some elements of their biology that is not directly related to this immunosignaling that's probably related to their morphology, yeah. Yep.
I guess one very quick, fast question here.
Two questions. First is, have you done this as a TED Talk? If not, you should. But my legitimate question is, what are the other sort of implications for human health and disease? I know you're here talking about IBD because of the topic of this forum, but have you thought about the rising sort of epidemic of food allergies or other sort of human diseases?
Yep, 100%. So you're thinking, so both with protists and with tough cells. So food allergy is another aspect that the lab's interested in. So a student in our lab, Gabe Barone, he's working, so one of the things that we found is that if you look at the follicular-associated epithelium, the thing that overlays the Peyer's patches where you have a lot of the antigen coming in, which is important both in defense but also in tolerance, tough cells, so these epithelium doesn't really have Panna cells, you don't really have as much goblet cells, you don't have enteroendocrine cells. You have M cells, and it turns out you have tough cells. And tough cells are important in actually shaping the biology of the Peyer's patch, and so we think that tough cells actually are playing a role in food allergy and food tolerance. And so that's an ongoing project. We haven't published anything on that, but yeah, we're working on that.
All right, thank you so much, Michael, again, for this very passionate talk. We all learned a lot about microbes and tough cells.
Yeah, thank you.
Okay, it's my pleasure again to introduce the third Michael of the morning. That's Dr. Michael Kattar from UCSF. He's like myself, an adult gastroenterologist. So Michael got his MD and his PhD here from Stanford, so he's very familiar with the campus. His lab is working on single-cell technologies as well as spatial omic technologies like MIBI or Codex to understand better how the interplay of immune cells and the epithelium and the epithelial injury in IBD can be like driving pathogenesis, disease activity, therapy response. So Michael, it's a pleasure to have you here today.
Okay, terrific. Well, thank you so much for the introduction and for this invitation. And it's really great to see such exciting work on mucosal immunity, IBD, and celiac disease. Here are my disclosures. And so I'll talk a little bit about hypothesis generation using single-cell multiomics to study inflammatory bowel disease and how we're profiling the effect of vedolizumab as kind of our initial campaign. And then the second part of the talk, I'll touch on accelerating the path to precision medicine using spatial multiomics of archived FFPE or formalin-fixed paraffin-embedded tissue. And in the last few minutes, I'll talk about hypothesis testing using patient-derived intestinal organoids to study the role of epithelial cells in IBD pathogenesis. So, of course, in inflammatory bowel disease, we now have 4 classes of medications for moderate to severe Crohn's, 5 classes for moderate to severe UC. all working by different mechanisms, and it's great to have many different options for patients, but the question quickly becomes, how do we choose the right therapy for each patient at the right time? And I think this case highlights why we need precision medicine for inflammatory bowel disease. So this is a 41-year-old gentleman with Crohn's of the colon and a colonic stricture. He tried oral medications for several years, had some benefit with an anti-TNF but lost response. So we tried a second anti-TNF with methotrexate, no response. We switched to anti-IL-12/23, he soon failed that. And at this point had really had years of suboptimal disease control, was intermittently on steroids. This is a picture on the bottom left here of the colon. You can see the ulcers and active inflammation. And then we tried vedolizumab or anti-integrin therapy, and he achieved deep remission within a few months. And these are the pictures here with mucosal healing and no active inflammation on the right. And so then the question is really, could we have somehow predicted this response to anti-integrin therapy and maybe saved him a few years of ineffective treatment? And so one way to approach this is to use multi-omics profiling in IBD, meaning integrating genomics, transcriptomics, proteomics, the metabolome, microbiome, et cetera. And these studies have highlighted pathogenic roles for stromal, epithelial, and immune compartments. And the goal is really to understand the immune system of each patient in depth in order to identify the most effective and least toxic treatment for each patient. And really, relatively few studies use simultaneous multi-omic methods on the same patient samples to enhance internal validity. So applying these methods to the same biopsies or the same blood draws, for example, and really approaching each patient as an N of one. And that's something that we're really motivated by. And so as our first kind of test case, we decided to look at vedolizumab and ulcerative colitis. Just to review, this is FDA approved for Crohn's and UC. This is a monoclonal antibody against alpha-4 beta-7 integrin. Alpha-4 beta-7 binds to MAdCAM1, which is a molecular addressant expressed on gut endothelial cells, and vedolizumab blocks intestinal migration of alpha-4 beta-7 positive circulating leukocytes. And so we wanted to use this to kind of optimize our multi-omic pipeline to study UC and the effects of vedolizumab. And there had been conflicting data on the primary cellular targets of vedolizumab, and I think most studies really emphasize the effect of vedolizumab on T-cell subsets. So that's sort of what we thought we would see as a reduction in certain T-cell populations in colon biopsies. And so we took a case control approach using multi-omics to understand the effects of vedolizumab. So we looked at healthy controls, UC patients on 5-ASA therapy, and UC patients on vedolizumab. We looked in the peripheral blood and intestinal biopsies or colonic biopsies. We performed biopsy site-seq, so this is a single-cell RNA sequencing, along with 187-plex surface marker profiling from both the biopsies and the blood. And then we performed CyTOF, or mass cytometry, looking at 37 surface markers on both cell suspensions, again, processed in parallel. And then we combined that with spatial transcriptomics and proteomics using some of the methods that I'll tell you a little bit about. And so I just want to emphasize the people that were involved in this work. So this was really led by Elvira Manillo, who's a specialist in the lab, as well as Kathy and Julian, who are CRCs and SRAs and former lab members. But we have a number of collaborators at UCSF and at the Chan Zuckerberg Biohub who helped do this work. And so we performed 187-plex site-seq on peripheral blood and biopsies. This is just kind of showing some of the data that you can generate using these approaches. So these are dot plots. For those who aren't familiar with this type of display, basically the size of the dot is the percent of cells that express that marker. We have the cell subsets on the rows, the genes on the columns on the left, and then on the right we have the proteins. Red is up, blue is down for the genes, and then brown is up and purple is down for the protein markers. Here I'm just showing at a very coarse level some of the landmark genes and proteins for each cell subset, but we have these for fine cell subsets as well, and this is really one of the first surface marker atlases for inflammatory bowel disease that we're aware of, and we're hoping to make all of the data publicly available very soon. But these markers and these kind of analyses informed our later spatial multiomics analysis, which I'll tell you a bit about. First, let me just show you some of the data from the site-seq. So these are the intestinal biopsy samples. This is 51 fine intestinal cell subsets. This is a UMAP here on the left, so each dot is a cell. The cells are clustered together by how transcriptionally similar they are. And we identified 51 intestinal cell subsets, and surprisingly, we saw a reduction in tissue myeloid dendritic cells associated with vedolizumab. And this was confirmed by CyTOF on matching samples. And this was surprising given that T cells are really frequently cited as the primary target of vedolizumab, but we saw this kind of outsized impact on dendritic cells. And then when we look in the CyTOF data, comparing the biopsies in the blood, we see a similar phenomenon. So this is a heat map on the left here. On the top, we're clustering the cells using, the patient samples using unsupervised clustering. In gray are the healthy controls, red the UC patients on 5-ASA therapy, and in blue are the patients on vedolizumab. And this heat map, again, red is up, blue is down, and this is showing the ratio of alpha-4 beta-7 positive cells in the biopsies versus the blood. And you can see that if you look across cell subsets in the patients on vedolizumab, there's a clear reduction, a relative reduction in the tissue and an increase in the circulation, really across immune subsets. So it does affect lots of different cell types, but because we have all of these data done simultaneously on the same patients, we can then look in the peripheral blood, and what we see is when we look at myeloid dendritic cells and the percent of alpha-4 beta-7 positive cells, when you have patients on vedo, the percent that are alpha-4 beta-7, percent of DCs that are alpha-4 beta-7 positive goes up from about 15% up to 80%. So these cells are really excluded from colonic tissue and kind of driven into the circulation by anti-alpha-4 beta-7. And so that suggests to us that DCs really may be more dependent on alpha-4 beta-7 for intestinal trafficking than other cell subsets. And this has now been validated in an additional cohort of 16 patients. And it just kind of highlights how you can use unbiased multiomics to identify new targets of current treatments. And so this is something that we're hoping to do for additional therapies. And so now I'll tell you a little bit about the spatial multiomics work that we're doing, also looking at vedolizumab. And so if you take a step back and kind of look at the state of biomarkers for precision medicine and IBD, we have examples of genotype, transcript, protein, autoantibodies. We have examples that incorporate the microbiome or multiomic signatures. And really, when you think about the barriers to implementing these clinically, we really only use genotype for the most part. There's been a lot of great work on precision medicine for IBD, but we're not really using a lot of these things in the clinic. And I think one barrier is that many of these methods require some specialized prospective sample collection, meaning you have to take a biopsy and put in RNA preservative or cryopreserve a biopsy, for example. And it's not difficult to do necessarily, but it's just not routinely done for every patient. And so in that regard, there would be a lot of advantages to using formalin-fixed paraffin-embedded tissue or FFPE tissue. This is, of course, the default tissue processing for every sample that we collect during endoscopy. We take a biopsy, we put it in formalin, that gets embedded in paraffin, it gets sectioned and reviewed by a pathologist. And so if you can take advantage of these samples, you immediately have access to longitudinal samples that are archived for years. Up to 10 years. And all of the cells that you're interested in are there. So for the work that I talked about earlier, where you cryopreserve a biopsy and then thaw it, digest it, and do 10x sequencing, you lose a lot of cell subsets there. Here, the cells are fixed, and so every cell type is there. And there are now methods to identify gene and protein signatures in FFPE biopsies with subcellular resolution, and I think that really has a lot of potential to accelerate discovery, development, and validation of biomarkers in IBD. And so we're using multiple different methods to do this, including multiplex ion beam imaging or MIBI, co-detection by indexing or codex, and RNA in situ hybridization methods. And I'm not going to talk about MIBI and codex because this is Stanford, and we have world experts like Stefan Regala and Michael Snyder is talking later, so I won't go into those methods in detail, but I'll tell you a little bit about some of the spatial transcriptomics work that we're doing. This is just an example of the CosMx method, where you're looking at 1,000 genes per cell with subcellular resolution. This is one representative image from a run here where each of the tiny dots that you can see are detection of one transcript in the cells, and then this is just kind of highlighting some of the key cell subsets in the colon. And so this was one pilot data set where we looked at 960 genes per cell in 16 FFPE biopsies from 10 patients, about 50,000 cells, pretty small data set, 17 cell phenotypes. But what you can see from this UMAP here, again, these cells are clustered together by how transcriptionally similar they are. This green cluster here, based on gene expression, looks like it would be cells at the top of the intestinal crypts. And I'm showing here on the right basically a dot plot with the landmark genes or the genes that are most enriched in each of the clusters. And in the blue cluster here, you kind of look like genes that would be enriched in the crypt base where the intestinal stem cells are. But the nice thing is then you can just click a button and say, where are these cells located in space? And in fact, these green cells are at the crypt top, the blue cells are at the crypt base, and then these orange cells are kind of the transit amplifying cells in between. And so you can go back and forth between sort of a UMAP versus a spatial scatter plot. And then the real power comes in, I think, when you can look and sort of gate at your cells of interest. So here we're looking at activated fibroblasts and inflammatory macrophages. So the fibroblasts are in yellow, the macrophages are in blue, and you can see that when you look in the ulcerative colitis sample, there's increased density and abundance of these cell subsets in active disease compared to healthy controls, and you can do kind of neighborhood enrichment analysis to kind of quantitate that. So kind of now that we knew the method worked, we decided to go straight to a retrospective longitudinal spatial analysis of archived clinical FFP biopsies. And so for these are just ulcerative colitis patient biopsies that are archived in pathology. These samples all ranged anywhere from 1 to 5 years old, not collected in any special way. We retrieved those samples and we looked before and after treatment in patients, with respect to vedolizumab therapy, and we stratified them as either responders or non-responders in addition to healthy controls. And we made tissue microarrays from these patients. So these are cores from FFP biopsies from these patients. This is just a subset of the tissue microarray that we analyzed. And then we looked at 1,000 genes per cell in this tissue microarray, as well as 64 proteins per cell with subcellular resolution. And then we basically compared transcriptionally, if you look before treatment in a responder or non-responder, what are the differences at the transcriptional level? And we can see a signature enriched in non-responders versus a signature that's enriched in responders. And that's shown in the heat map here. And because it's single cell, we can say, okay, on a bulk level, we can identify this gene signature that's enriched in responders or non-responders. Where are these expressed? And if you look at the non-response genes, the genes that were most associated with vedolizumab non-response, those were expressed in activated fibroblasts and macrophages, whereas the response signatures were really expressed preferentially in the crypt base, where the stem cells are. And then we can go further, and so that was looking at single cell, but then if we look where are these cells and genes in space, here I'm showing some representative areas from the tissue microarray. So we have the healthy controls, pre-treatment vedolizumab responder and non-responder, and then post-treatment. These kind of small green lines that I know are hard to make out are the cell pose analysis of basically the cell segmentation, looking at the borders of each cell. And then I'm showing below that the cell types. So in gray are the epithelial cells, and yellow are activated fibroblasts, and blue the macrophages. And you can see that before treatment, those cells seem to be more abundant. And then post-treatment, they are reduced in responders, and they go up in non-responders. And then we can look at those genes that were associated with vedolizumab non-response, and they seem to co-localize with those cell subsets. And so basically, this suggests that activated macrophages and fibroblasts are associated with vedolizumab non-response. And in contrast, when you look at the response signature, those are really enriched in the epithelial crypt base. So the crypt base here is shown in pink, crypt top is in magenta, and then these class 2 high epithelial cells are shown in red. Those are a little bit more rare. But then when we look at the genes that were associated with vedolizumab response, those seem to overlap with those intestinal cell subsets. And so this really suggests a potential mechanism for mucosal healing, that when you reduce inflammation with vedolizumab, that the patients that are the most likely to respond are those that kind of have a more robust epithelial response and are able to achieve mucosal healing. And this was validated in an external bulk transcriptomic data set. And so now we're starting to look at additional therapies and see if we could predict prospectively using scores kind of like this, who would respond to which therapy using archived FFPE tissue. And so in the last couple minutes, I'll just mention a little bit about some hypothesis testing studies we're doing using patient-derived organoids to study epithelial cells in IBD. And so just to review, IBD is the result of an abnormal immune response to microbial stimuli in genetically susceptible individuals occurring in the right environmental context, and that culminates in epithelial injury. And currently, all of our FDA-approved biologics target the immune system in some capacity, and that makes sense because IBD is an inflammatory disorder. At least as of right now, we have nothing that targets the microbiome or the epithelium, although hopefully that will change in the future. And so our hope is that if we study some of the genetic factors and the microbial factors that regulate epithelial cell death, that will help us identify non-immunosuppressive or less immunosuppressive cytoprotective approaches in IBD, and that's something that we're really excited about in terms of using intestinal organoids. And so intestinal organoids really have a lot of potential in terms of studying IBD and understanding IBD. And just to review, to grow these, we take biopsies from patients. Those biopsies contain intestinal crypts. The crypts contain intestinal stem cells. We embed these in an extracellular matrix and supplement them with growth factors, and they form these 3D structures in the lab, and that allows us to study epithelial cells. They grow, more or less indefinitely. You can freeze and thaw them, and they recapitulate many aspects of epithelial cell biology, and they allow us to perturb living cells in vitro. I mean, everything I talked about before, I think there's a lot of power in profiling, but it's really a snapshot of the immune system for each patient. Whereas here we have living cells that we can stimulate and see how they react, and that can be really beneficial for understanding biology. So in terms of guiding therapy, organoids are really closest to the clinic, I think, for cystic fibrosis and for cancer, but there are some really nice examples in inflammatory bowel disease. This is probably my favorite example, actually, from Alexa Muse and colleagues looking at a patient with a TTC7A mutation that had very early onset IBD, increased epithelial apoptosis, severe enterocolitis. They did a drug screen and found that luflunomide rescue organoid death in vitro, and then they used that off-label in that patient, and the patient improved clinically. So really a very powerful bench-to-bedside story. And there are other examples using patient-derived organoids to study responses to inflammatory cytokines, and a lot of ways to think about incorporating patient-derived organoids into understanding IBD. I'm going to provide two very quick examples of what we've used them for in the lab. So we're using them to identify new cytotoxic factors. So we found a role for lymphotoxin-induced epithelial death. Lymphotoxin is a cousin of TNF. It's a TNF superfamily member. And we saw that it actually exhibited similar cytotoxicity to primary epithelial organoids as TNF did. And so there may be some patients that benefit from lymphotoxin blockade in addition to TNF, and that's something we're still investigating. We're also interested in using organoids to find rare responders to therapy. So this is one example looking at GSK963, which is a RIP1 kinase inhibitor. RIP1 kinase is a signaling protein that's involved in death signaling in intestinal epithelial cells. It actually failed phase 2 clinical trials for ulcerative colitis, but we found, looking in a panel of organoids from non-IBD and IBD patients, that there was one patient that benefited in vitro from RIP1 kinase blockade in the presence of TNF. And so it's just kind of proof of concept of how you could use patient-derived intestinal organoids to find rare responders to therapies that don't work for all patients. There are other studies, looking at JAK inhibition to block TNF or interferon gamma-induced injury. So really lots of potential ways you could think of incorporating patient-derived organoids for precision medicine. And there are actually some companies now looking at augmenting healing using IL-22 or Wnt surrogates, and I think these organoids will really be a nice way to test to see who would respond best to those types of approaches. The last thing I'll show you is one piece of data that we've generated recently. So we are now basically performing matched single-cell RNA sequencing of epithelial cells at the time of collection, and then we're making organoids from those same patients expanded in parallel, and then we sequence those as well. And I'm showing on top a UMAP of the intestinal epithelial cells analyzed by single-cell RNA sequencing. Each different color is a different patient, and you can see that the cells are really distributed on the top and kind of intermingled, whereas when you look at the patient-derived intestinal organoids, they really cluster by patient. And so there's a lot more patient heterogeneity that we see in the organoids than we see immediately ex vivo. And that's something that we're planning to investigate more. This is work in progress, but we have 30 organoid lines where we've done this that are kind of really well characterized in a serial manner like this. And I think that'll provide a nice opportunity to explore the epithelial contribution to IBD. So just to summarize, single-cell and spatial multiomics are providing insights into specific therapies in IBD. Clinical FFPE samples are compatible with emerging spatial multiomic methods. Studying epithelial cells, I think, could help identify less immunosuppressive approaches. Working with patient-derived organoids can help understand the epithelial response to injury. And we're also expanding our co-culture methods in terms of co-culturing with immune cells, fibroblasts, endothelial cells. So I think that'll be really exciting. And then we have some studies now starting to look at pediatric IBD, some early onset, really refractory patients with Alice Chan, Sophia Verstraete, and Sujin Cho, and hopefully we'll have data that we can share with that soon. And so I'll just take a moment to acknowledge the folks in my lab who did this work, our GI colleagues, all the participating patients, our collaborators, and our sources of funding, and happy to take any questions.
Thank you very much, Michael. Very nice talk. Are there questions? I guess there are questions. Yeah.
Thank you so much for the fascinating talk. So in practicality, what we see sometimes is that patients who've been treated with anti-TNF agents don't respond that well to vedolizumab.
Yeah.
Do you see any genetic changes in
Yeah.
those patients which make them poorly responsive to vedolizumab?
Yeah, it's a great question. We did look at that, and basically our non-response signature for vedolizumab also was associated with non-response to infliximab, but that's because, I think partly because all of our vedo non-responders had already failed anti-TNF, and the publicly available data, that's true too, that all the vedo non-responders had failed anti-TNF. So it sort of, in a way, is a validation that that non-response is sort of universal to both. But the response signature that we found for vedolizumab was unique to vedolizumab. It didn't predict infliximab response. So that could be something where you say, okay, this person is more likely to respond to vedo and less likely to respond to infliximab, that kind of thing. So.
Michael, in full agreement, N=1 is the future.
Yeah.
And also that there is not a way but personalized intervention using organoids in a mucosal inflammatory disease like IBD. But this is not now, it's going to be in the next generation because we have no drugs that go after mucosal injury and homeostasis. Yeah. But I know that this is going to happen. I don't know when, but it's going to happen. But right now, you were talking about possible roadblocks to really use technology that we have in hand. And I'm wondering why single cell or spatial transcriptomic analysis unbiased compared to what can be done in a cost-effective way. Take all the drugs on the market approved for IBD and focus exclusively on pathways that they go after, and you do target, bias analysis and find the best drug until, again, the holy grail of the mucosal injury will come to fruition.
Yeah, that's a great idea. So you're saying favoring doing more biased approaches where there are already approved compounds and approved methods. Yeah, I think that's a great idea, and I think that makes the most sense. I feel like in Europe that's starting to happen with some of the organoid work. I think that my understanding is there are some situations where they'll approve a drug, at least for cystic fibrosis, for example, if you can show that the patient-derived organoids, there's a benefit. But in the US, I don't know, it seems like that is still a ways off. But I agree with you. I think biased approaches make a lot of sense, more so than these large unsupervised approaches, which are more expensive. Yeah, absolutely.
Are there other questions in the audience? Yeah.
Just a quick question. For the myeloid DC changes, did you think those were direct or indirect, or do you know?
The reason, it's a great question. I think the reason we think it's direct, I think if we were looking at something like anti-TNF or anti-1223, maybe it's indirect, but because they're alpha-4 beta-7 positive, and then we're treating with anti-alpha-4 beta-7, and those are the cells that are now suddenly, 80% in the circulation as a pre-treatment, they're kind of like 10%. That to me seems like a direct effect. But you're right, we can't, that's the one big limitation of these sort of descriptive methods, and why I think we still need mouse models, so.
Any additional questions?
Yeah, hey. Thank you, Mike. I thought it was, getting back to what Alessia was talking about, I think it's very interesting that your response signature to vedo was an epithelial one.
Yeah.
And so does that suggest that it's not just enough to take away the immune response, and that there are some folks that don't have that response signature, and there is a place for epithelial-directed therapy?
Oh yeah, I think for sure there's a place for augmenting that response. Like, and some of those genes are sort of downstream of Notch or Wnt signaling, so you could imagine a scenario where you block inflammation and at least transiently try and help the epithelium recovery with like a Wnt surrogate or a Notch ligand or something like that. I think that's a great idea. Yeah, absolutely.
We just have to convince people it won't cause cancer.
Yeah, exactly. That's right. Yeah. For induction, I think it could work. Maybe maintenance, maybe not so much, but good point.
If there's, if I have a second question too. So maybe costs will come down eventually on these things. So taking that's obviously a hurdle in terms of scaling this.
Yeah.
you mentioned like traditional single cell approaches, like I'm not totally convinced even that when you cryopreserve them in a certain way, you can really dissociate all the cells and get all the information you would need as if you would do it fresh, but it's impossible to scale fresh. There's spatial, there's single nuke. Have you thought about if you could do, 100 and you had to scale across multiple institutions? Like what would be, is there a path forward to that? Or does
I
this
think
really
so.
have to be a small?
I mean, I think these new spatial methods, some work better than others. And I think some of the custom panels, which I didn't have a chance to show because we haven't gotten that kind of ready to present yet, but you can really drill down on all of these cell subsets pretty well. And then you have all the cells intact that you're interested in, like you're not losing epithelial cells. We don't see any neutrophils by 10x with the freeze thaw. Here with the spatial, we're able to see all those cell subsets and where they are in space. And I think that can scale. And if the cost is always a barrier, but if you fit a lot of samples onto a slide, it's actually cheaper than single cell sequencing. So I think, doing hundreds of patients is possible. Yeah.
So that the microarray approach that you took is a way to reduce costs.
Yeah, it reduces cost significantly. Yeah, that's right.
Do these multiplexed in situs overcome issues of degradation with FFP that when you tried?
That's a good question. We have not looked in any samples that are older than 5 years, and there is some variability for sure in tissue quality, but these are at least 5 years old, these samples, and we didn't really see a correlation with age or a big difference between samples we had prospectively collected with FFPE and processed on our own as opposed to what's kind of been sitting in pathology. So it was overall, I think, very encouraging for us that this will be useful, going forward. Yeah.
Thank you.
Any additional questions? All right.
Thanks
Thank you so much,
yeah.
Michael. So I guess with that, we're closing session 1 here. So enjoy the break, and thanks again to the speakers.
if we could all take our seats, we're ready for the next section. Pretty close. Okay. Okay, so thank you all for sitting down and getting ready for our next section on celiac disease. Our first speaker is Hilary Jericho, and Dr. Jericho is a clinical associate professor of pediatric GI at Stanford, and as you heard from Michael before, she's the inaugural medical director of the celiac disease program at the center, which we're all very excited about. She's an expert in pediatric celiac disease, and she has a particular interest in clinical management and in causes of refractory disease. She's been a frequent speaker at national and international celiac meetings, and she'll speak to us today on the psychosocial and financial impact of celiac disease. Hilary.
Let's see. All right. Oh, it is picking up. All right. Well, thank you, Betsy, so much for that introduction, and thank you for having me here today. It's a privilege and an honor. As Betsy mentioned, I'll be speaking on the psychosocial and financial impact of celiac disease. I have no disclosures. So my objectives today are to improve the understanding of the most common celiac presentations in the course of symptom resolution, discuss the psychosocial impact of celiac disease, and learn more about the financial impact of celiac disease. All right, so this will be very, very brief because I'm sure people are extremely familiar with celiac disease in this crowd. But just a quick recap, celiac disease is an autoimmune enteropathy in genetically susceptible individuals, and it's triggered by the ingestion of gluten, which is the major storage protein in 3 grains, wheat, rye, and barley. It will cause a variety of inflammatory damage to the mucosa of the small intestine and then can result in a variety of clinical presentations both within and outside of the GI tract. As we know, the only current available treatment is strict adherence to the gluten-free diet. And if it's left untreated, it can lead to systemic complications, including increased mortality. And it's thought to affect about 1% of the general population, though many of us think this number is actually higher than this. All right, so I'm going to start early in my career, what kind of got me down this path of looking at the psychosocial and financial impact of celiac disease. One of the first studies I did at the University of Chicago was looking at the extraintestinal manifestations of celiac disease and the effectiveness of the gluten-free diet in these symptoms over time. So this was our study group that we looked at. We had 737 patients at that time with confirmed celiac disease in our database. Of these, 347 patients were children, 390 were adults. 209 of the children had extraintestinal symptoms and 244 of the adults. And ultimately, 157 of the children and 172 of the adults were included into our study. So what we looked at were the most common extraintestinal manifestations. And while we came across all of these symptoms in our patients, the symptoms that we saw at the highest rates in the P is pediatrics, the A is adults. So the 4 most common symptoms that we saw in pediatrics was fatigue, headache, poor mood, and short stature. And the 4 most common symptoms, extraintestinal symptoms that we saw in adults, a lot of them were overlaps, including fatigue, headaches, anemia, and then psych disorders. So in both groups, we did see that there was some mood or psych component that was a higher manifestation. Here, what I want to show and demonstrate, use the laser pointer, we were looking at the rates of improvement over time of these extraintestinal manifestations. First, I'm going to show you a graph of children, and second, I'll show you the graph of adults. while this captures all the symptoms, and you can see that this is the percent, 100% existed at the baseline, and then this is over time up to 24 months, and how these symptoms improved over time. And as a whole, children had very good rates of improvement in symptoms. Some had complete resolution of some of these symptoms by 6 to 12 months, the vast majority by 24 months. But what I tried to highlight here specifically, this is rates of improvement of poor mood, and this is the psych disorders. And as you can see, some of the worst rates of improvement in pediatrics were of these psych disorders, even out to 24 months on strict adherence of the gluten-free diet. Next, we're going to look at adults, same setup. But as you can see, for the mood disorder and psych disorder in adults, as a whole, rates of improvement were not nearly as good. We didn't see complete resolution in any of the symptoms till out to 24 months, so it took much longer in adults. And then those rates of improvement for mood disorders and psych disorders were, again, among the worst rates of resolution in adults as well, which got me more interested in this topic to look further into it. So ultimately, the conclusions of this study were that children and adults with celiac disease, they have similar rates of extraintestinal manifestations. In children, short stature, fatigue, headache, and poor mood were the most common symptoms. In adults, anemia, fatigue, headache, and psych disorders were the most common. Children on a strict gluten-free diet did show faster and higher rates of resolution as compared to adults. But in both groups, psych disorders and poor mood showed some of the worst rates of resolution over time. So this brought me into a follow-up study looking specifically at rates of depression in our pediatric population and its impact on quality of life in these individuals. And just to jump right into the results of this study for the sake of time, what we're looking at here, there's two different charts, one on a descriptive analysis of different results we found, and then associations. And ultimately, the age at the time of the survey of the majority of our patients, the mean was 14 years of age. Most of them had been diagnosed around 10 years of age, so they were about 3 to 4 years into their diagnosis. The depression scores that we saw at 13 and 12 were actually quite impressive. These dropped these individuals into a moderate depression category that was comparable to those of adolescents with known mental health disorders in a psychiatric clinic sample. And on average, those scores are about twice as high as the average adolescent in the community, looking at a population of white and black adolescent females. In addition, the adjustment to celiac and the PEDS quality of life scores are also pretty poor. This is a scale of 0 to 100, so scores of 27, 29, and 41 really are not very good and are a bit concerning. We wanted to then look again on how all these different factors associated both with quality of life and adherence to the gluten-free diet. As a whole, we didn't find there was an association between age at the time of the survey, age at the time of diagnosis, years on the diet, adjustment to celiac in the youth, or symptoms. There was no association between those and quality of life in these individuals, but there was a very high association between depression scores, both from children and their parents, and the parental adjustment to celiac disease and that impact on quality of life. We were ultimately looking at how these variables affected adherence to the gluten-free diet, and while there was no statistically significant association, it was approaching statistical significance. So our conclusions in this study were that adolescents with celiac disease reported levels of depression that were comparable to those reported by adolescents seeking mental health services. The length of time living with celiac disease on a gluten-free diet, the age at diagnosis, and the perception of the disease did not appear to contribute to the depression. The high rates of depression, we were concerned, could ultimately impact celiac disease prognosis and adherence to the gluten-free diet. Therefore, screening in this age group is of utmost importance. And identification and intervention for that depression could lead to improved adherence of gluten-free diet into adulthood. So next I want to switch gears a little bit and talk about the financial side of things and then tie everything together. So I'm turning to a study entitled Food Insecurity insecurity negatively impacts gluten avoidance and nutritional intake in patients with celiac disease. This was a study by Ma et al., and what they did is they used the NHANES survey from 2009 to 2014. The inclusion criteria were all participants with celiac disease who completed this SFSS module, and patients that were excluded were predominantly anyone who failed to complete the survey, had incomplete dietary records, were missing data, et cetera. And what this was looking at was the proportion of patients with celiac disease that were living with food insecurity. And then they wanted to compare the effects of food insecurity on how that impacted those individuals' ability to adopt the gluten-free diet, and also their micronutrient and their macronutrient consumption. And these are the survey results, or the study results. So as a whole, they had, just over about 2,700 patients or so. Of all those patients, everyone in this category were patients affected by food insecurity, and these are the individuals who were food secure. There were 454 celiac patients that were affected by food insecurity. That is 16% of their total population of celiac patients. And what they found is that 66% of these individuals were female, and that the people most impacted here were those of a slightly younger age. They were non-white individuals, they had a lower educational background, and they were living in poverty. What they show over here in this graph is that as a whole, these are individuals, again, in food insecure houses versus food secure homes. The amount of micronutrients and macronutrients taken in by these individuals across the board, every category, was lower than the RDA for those individuals as compared to those who were in food secure houses, with many of these reaching statistical significance. So the ultimate conclusions of this study, as I pointed out some of these already, 16% of patients with celiac disease in the United States are affected by food insecurity. Food insecure individuals with celiac disease were disproportionately younger, poorly educated, non-white, and living in poverty. Food insecurity was associated with significantly lower consumption of protein, carbohydrates, fat, and most vitamins and minerals. And the last aspect of the study that I didn't point out on the last slide that they looked at, again, was how did this impact adherence to the gluten-free diet? And what they found is that a significantly lower proportion of individuals affected by food insecurity who had celiac were able to adopt the diet, with only 24% adherent to a gluten-free diet when affected by food insecurity, as compared to roughly 68% of those who are not affected by food insecurity. Moving along to the financial impact of the gluten-free diet in the treatment of celiac disease. So I just want to do a quick refresher again, since we've moved into this presentation away from the first slide, just to remind everybody. So again, celiac disease is an autoimmune enteropathy in genetically susceptible individuals triggered by the ingestion of gluten. The only current available treatment for celiac disease is strict adherence to the gluten-free diet, which I think, as many of us know, can be very expensive and difficult to adhere to. And unlike other disease conditions out there, for celiac disease, families bear 100% of that cost of the treatment, without the ability to use FSA, HSA spending, insurance reimbursement, coverage, or tax deductions. And this is partially true. I'm going to get into how this is laid out in the US and who is eligible. So I'm going to turn to another study first, entitled Persistent Economic Burden of the Gluten-Free Diet. And again, I don't think that this comes as much of a surprise to anybody, but what this chart here is showing, and this is a study by Lee et al. In 2019, is that these are all starch-based items that obviously can come in a gluten-free or gluten-containing version. And the blue bars are all your gluten-free options, and the red bars are all your gluten-containing options. And it's pretty clear that across the board, this is the cost of these items in US dollars compared to the items we're looking at. The cost of those gluten-free foods is substantially higher than those of the gluten-containing counterparts. And in general, gluten-free products are roughly 242% more expensive than their gluten-containing counterparts. This is just to look at across the US location as well. Well, maybe the first study, it was one city and things are disproportionate. But this also looks at across the US a number of different cities: New York, Chicago, Atlanta, Rapid City, which is South Dakota, in case anybody doesn't know, because I didn't actually know where that was, Portland, and then even online. And across all these regions of the US, again, gluten-containing or gluten-free foods are substantially more expensive than their gluten-containing counterparts. So the last thing I wanted to look at, okay, well, we know they're more expensive, but what is the actual cost that these families are bearing each year? And maybe I just did not find the study, but I was trying to find a study that had that information, and for the life of me, I couldn't find anything out there. So I turned to a very good friend of mine and a dietitian at the University of Chicago who I used to work with, and she has celiac, her son is celiac, and on average they spend about $300 more per month than on those gluten-free foods than they would if they weren't buying them, which averages about $3,600 extra per year that they are spending. So I just wanted price point comparison. This is celiac IBD here. I thought, okay, they're spending $3,600 a year out of pocket on the gluten-free diet. What is insurance covering for our Crohn's patients for the cost of a pharmaceutical medication? And I think this probably comes as a shock to nobody. I brought up a chart of biologics, which at our cheapest these days is Inflectra, costing about $16,000 per year, ranging as high as Remicade on the scale at about $46,000 per year. So insurance is covering much heftier bills for the treatment of Crohn's disease than they would be for the gluten-free diet, which is, again, the only quote-unquote medication for celiac disease. So I just want to very, very quickly go through what system does exist, because we do have a system to cover the cost of the gluten-free diet for patients with celiac disease. I will not say that we do not. 4 very, very easy steps to get medical tax deductions. So step 1, not bad. You just have to get a letter written from your physician and a prescription that you need to be on the gluten-free diet. You're going to need to submit that with your tax forms. 2, you have to save all detailed receipts of those gluten-free groceries, not just one time and extrapolate out to a year, all of them for the entire year. And then what can you deduct? Well, you can't actually deduct the full cost of that gluten-free food. You can deduct what's in excess of what you would have paid for the gluten-containing counterpart. So you need to do that calculation, and not only do you need to do that calculation, you have to have proof of what that other food item costs. So now you're not only buying gluten-free foods, you're buying the gluten-containing counterpart to show objectively what that cost difference is. If there's anything specific to a gluten-free diet like xanthan gum, you can deduct that full cost, and then cost of travel and any shipping if you're ordering things online. All right, so step 3, as if this is not now getting tedious enough, you have to then determine at the end of the year all the items that you can deduct. You have to make all those calculations. Hopefully you have a spreadsheet, because you're probably looking at a lot of calculations, and then you need to submit all of your receipts. Last but not least, as if that wasn't tedious enough, you get to fill out this lovely, lovely form. All we need is another tax form. And that is the 4 easy steps to submit the cost for your gluten-free diet. But we're not done yet. So the caveat, Section 213 of the Internal Revenue Code provides an itemized deduction for qualified medical expenses that are incurred, but under the present law, the medical expenses that you can deduct must exceed 10% of your adjusted gross income. So what that means, for example, I'm just throwing a random number out there, if your adjusted gross income, you make $100,000 a year, that means you can't start deducting the cost of that gluten-free diet until it exceeds $10,000. So going back to my dietitian and her expenses, so she's spending $3,600 a year on the gluten-free diet, which, you know, for some families that's doable. There are a lot of families out there that is a very big cost. And so in order for her to be able to submit anything, the max she could make to be right at that number would be $36,000 a year of salary. So you can see, not only is it arduous, not a lot of people will even qualify for that. FSA, HSA plans, it's more or less the same policy. So I'm going to wrap up on this last portion. I'll just spend about 5 minutes here and then open up for question and answers. So this is a study underway right now that myself, Ritu Verma, and Kate Keenan, who's a PhD at the University of Chicago, we've done a lot of studies together. We got a grant to do the study. We're towards the end of the enrollment process, and then we'll analyze the data. But we're looking at the psychosocial and financial impact of the gluten-free diet in the treatment of celiac disease. So we've got a lot of objectives here. The first one is to calculate that incremental cost per family per month of purchasing that gluten-free diet. The secondary objective is to then take that cost and be able to extrapolate it out to understand what would the annual cost be. The tertiary objective is to compare that cost to the cost that's being covered for other chronic illnesses. Our quaternary cost is to also assess the psychosocial impact that the gluten-free diet is having on these families through standardized surveys looking at perceived stress and other surveys looking at food insecurity. And then our ultimate goal is to work to reform the current system for medical insurance and tax reimbursement to get a system in place that is going to work for our families with celiac disease. So this is a long methods. I won't go through every single aspect of this, but again, it's a multi-center study between us here at Stanford and the University of Chicago. With the help of a biostatistician, it was decided we needed 32 new families. They're doing a number of surveys, again, looking at demographics. We're collecting grocery bills, and then we're doing standardized surveys looking at the financial, emotional, and social impacts of being on the gluten-free diet. We'll collect all itemized receipts, again, to be able to extrapolate what is the annual cost on average to families who are on a gluten-free diet. People are getting compensated for their time, and then ultimately the hope is to advocate for a legislative measure designed to reform the current medical insurance reimbursement system to make the system simpler and more doable for our families who have celiac on the gluten-free diet. So last but not least, is this feasible? So there are currently bills in place that do allow for coverage of nutritional medical treatment services, though everything that's in place right now, unfortunately, only revolves around diabetes or kidney disease or post-kidney transplant with a physician referral. There is a situation similar to celiac disease where reform did occur, and that is on the topic of cow's milk protein allergies, which affects about 2 to 6% of children. As many of us know, that does require strict adherence to a cow's milk protein-free diet. And in 2012, after significant advocacy highlighted the heightened cost and the necessity of being on that cow's milk protein-free diet for children with cow's milk protein allergies, fully insured plans did become subject to state and federal law that required mandates or had mandates on insurance requiring coverage for those elemental formulas for patients with a cow's milk protein allergy. So in summary, psychiatric and mood disorders are prevalent in both pediatric and adult celiac patients and often exhibit limited improvement when managed with a gluten-free diet. There's social factors that contribute heavily to the high rates of psych disorders that include things such as limited food choices, cross-contamination, dietary restrictions, social events, social isolation, the symptoms, the financial cost, and food insecurity. Although insurance often will cover costly pharmaceuticals, there is currently no easy mechanism in place to provide coverage for the only treatment for celiac disease, which is the gluten-free diet. So ensuring equitable care for individuals with celiac disease is going to necessitate a revamp of the existing system, addressing the financial burden that's borne by celiac families due to that additional cost associated with the gluten-free diet. Thank you very much. And I'm happy to take any questions.
Yeah.
Thank you, Hillary. I think this was a wonderful and very enlightening talk. I have some questions about the psychiatric disorders that you screened for. Were you specifically looking for just anxiety and depression, or did you extend it to other things like ARFID and other psych disorders? And second question is, I know you looked at the age of diagnosis, but typically what I see is that children who are diagnosed at a very young age, I would say under 5 years of age, tend to adapt better than patients who are diagnosed at teenage years. So could you please give us some differentiation among those two groups?
Yeah, no, those are excellent questions. So most of the surveys that we had used in Chicago on that study was the CDDUX and the PROMIS, and most of these did look at more anxiety, depression, as opposed to eating disorders and other psych-related issues. And then we had some additional surveys looking at, again, quality of life, and they were all adolescents, so it was a very biased set of results that we got for the adolescent population. We did look at it both from the parental perspective and the adolescent perspective, though, to ensure there wasn't any bias there. But what we focused on predominantly was looking at anxiety and depression. But I agree with you. I think there's many other factors that go into that could be studied further. And then to answer your second question about adaptation, I mean, just from a personal perspective, I do agree with you. I think, and I think a lot of studies have looked at this too, that it is true. I think the younger, and for many chronic illnesses, the younger the child is at the time of diagnosis, I think they often do adapt better because that is the life they have known, versus when you're used to a certain lifestyle and then it abruptly changes, especially in a time of your life when you're so vulnerable, such as adolescence, and there's so much changing. So that was one of the main reasons we actually picked the adolescent population to look at, because they were so vulnerable and they're at such high risk. But I agree with you. I think the younger individuals are across many chronic disease entities. When they're diagnosed, they do adapt better than diagnoses in adolescence.
Yeah.
With the food manufacturers to get them to say, okay, our Trader Joe's gluten-free product is this much, and the comparable non-gluten-free product is this much. Would that help the families keep records better?
Yeah, no, I think that's a great question, and something we're actually doing, and I think this would make it easier. So many of the big chains now actually have apps. And this is how we are getting these itemized bills from people, is we're having them download the app. So these apps will keep track of your receipts, and they'll itemize them, and they will pull out costs, which has revolutionized the system greatly. And it probably would make it easier for individuals, I think, to be able to submit. But I think at the end of the day, again, having to make all those steps, maybe it's doable for adults, but I think, in my opinion, having two young children, a 9 and 11-year-old, if I was asked to do that, I just do not see that I would have the time and energy to sit there and collect all these bills, even if it was electronically done for me, and then make all the calculations and have the spreadsheet. So my vision is, just understanding on average what is that additional cost these families are taking on by adhering to a gluten-free diet, and allowing some system in place that they just get that lump sum of money each year, or they're able to put it through FSA or HSA and not have to go through that tedious process of all of these calculations.
I mean, I think by having the manufacturers really delineate the differences may put pressure on them to try to minimize the differences. It's another hope.
Yeah, I think, it's all about I would say demand and quality of items used, and unfortunately a lot of those that go into making gluten-free products are more expensive, and there's just far less competition out there to drive down the prices. And so it's a tough situation. There's certainly like Aldi's and Trader Joe's, I agree with you, they have wonderful options and they are more affordable, but it's still a higher cost. And for families that are just scraping by, even that little cost might be too much. So yeah.
So we have time for one more?
So, thank you so much. So it's like a two-part question. The first is, in your initial study, you differentiated between psychiatric disorders and low mood. So there was a distinction right there, and I wanted to hear a little more about that and why there was a difference. And the second is, in terms of advocacy, what is the organization that you feel would make a big impact on helping legislation?
Yeah, so I'm trying to remember back. That the first study was over 10 years ago now that we started that one. And I'm trying to remember, it was a chart review or, we pulled all the patients from the database and then did a chart review to see. And I think it was just how they were classified, that some were truly had the diagnosis of anxiety or depression, and then some people in the chart review were just listed as low mood, and because it wasn't an official anxiety depression, that's why we separated them, just because we didn't know if it was formal diagnoses, which then led to those two different classifications. And then with regards to advocacy, you're asking how I intend to move forward with that?
Yeah, so what is the organization that you feel would be helpful, to legislate and, be able to reach assemblymen?
Yeah, so as I'm sure with many of us, getting into that field is very out of my expertise. So my hope was to call upon one of the lovely things about being at an academic center, such as Stanford or many of the other centers represented here, is we have many graduate schools. And so I'm hoping to recruit from the law school who have the expertise needed to pursue that and work together in a collaboration to make change.
Okay, so we want to thank Hillary for this wonderful talk, and we'll move on. I have this mic? Okay, great, thanks. So we'll now move on to our keynote address, and our speaker is Dr. Alessio Fasano. He holds the W. Allen Walker Chair of Pediatric GI and Nutrition at Mass General, and he's also the chief of the Division of Pediatric GI there. And he's the director of the Mucosal Immunology and Biology Research Center, again at MGH. He's an expert in celiac disease, intestinal permeability, and autoimmune disorders. And his research team established the rate of celiac disease as 1 in 133 people, leading to the awareness of celiac disease as a growing public health problem in the US. Today, he's going to speak to us on the importance of adequate nutrition and the intestinal microbiota in the balance between health and disease early in life, the celiac disease paradigm. Thank you.
All right, thank you, Elizabeth, for the kind introduction and for, of course, the invitation to be one of the keynote speakers today. Again, what I'm going to try to do in my next 35 minutes is to really touch upon what Hillary just mentioned and the Michaels, present and future, Michael Snyder as well, because a lot of stuff that you hear about celiac disease applies to IBD as well. These are my conflicts. This is almost exclusively related to diagnostic and therapeutic exploitation of the microbiome. We're not going to discuss too much about this today. Rather, what I'd like to do is, discuss, as Michael touched upon, about the education of the microbiome and the immune system, particularly in the first 10,000 days of life. We will outline the interconnection of 3 key elements that break tolerance. One is the dysbiosis, the other one is the immune system and the antigen trafficking, and how this really affects the balance between health and disease, and then provide celiac disease as a paradigm that to cure can apply to IBD. And last and not least, what is the future in terms of changing the history of this situation to try to modulate through nutrition the entire deal. And I'd like to start with a slide that probably is used and abused. That is the hygiene hypothesis. Just to put this in the context, we as a species, for almost the totality of the 2 million years of evolution, we got sick and died for mainly one reason: infections. And this was until the very, very recent past, the last second of this evolutionary journey, when we start to really have an understanding of bacterial pathogenesis, infectious disease pathogenesis in general. And in the Western Hemisphere, people that embrace a Western lifestyle, we start to implement measures like sanification of the water, antibiotics, vaccine, and so on and so forth. And with that, we saw, again, who embraced that lifestyle, a plummeting of infections like TB, mumps, Hep A, and rheumatic fever, and so on and so forth. But during the same period of time, we saw this equally impressive, but the upward increase on non-infective chronic inflammatory diseases, including, like IBD and autoimmune diseases, including celiac disease, neurodegenerative diseases, cancer, and so on and so forth. So in other words, if we embrace a Western lifestyle, we don't die fast of infections. We die very slowly of chronic inflammatory disease until Mother Nature decided to throw a curveball with the pandemics or with SARS-CoV-2, and everything starts again. At that time, the paradigm, the two pillars, why we got healthy or we are susceptible to disease were based on two pillars: who you are genetically, so your genetic predisposition, and the environmental exposure to triggers of inflammation. This timeline cannot blame genetic mutation. That takes much, much longer. So the conclusion was, okay, we're changing the environment too fast for us to adapt, and we pay a price if we embrace a Western lifestyle. That's definitely, a way to interpret this epidemiological phenomenon in a relatively pessimistic way. But if you want to see the glass half full, there's another message there that we learn, meaning the old paradigm, one gene, one protein, one disease that led to the Human Genome Project and so on and so forth, that implied if I'm genetically predisposed for Alzheimer or colon cancer, there is not much I can do about this. This epidemiological phenomenon suggests something differently. Suggests that that's not true. It's not a given that if I'm genetically predisposed, I will come down with that problem. It depends how I play my genetic cards. So in other words, my lifestyle. And I believe, as we chat with Michael as well, the future of personalized medicine is to understand what we've been doing wrong by embracing a Western lifestyle, try to put in place remedies to mitigate that upward phenomenon, if not completely reverse. And that led us a while ago, and that was not totally accepted until the recent past, to a different way of reading this, meaning that, yes, genetic predisposition and environmental triggers are necessary, but not sufficient. There are at least another three elements that seems to be at play that will dictate our clinical destiny. These two worlds are normally segregated by barriers. The most visible is the skin. The largest and the most complex is the GI tract. So there is a lot of literature of impairment of the barrier that allows antigen trafficking and instigate the immune system to generate inflammation. Of course, the fourth element got to be the immune system because we're talking about chronic inflammatory disease. So an immune system becomes hyperbelligerent. And last but definitely not least, this ecosystem Michael was mentioned that is mainly focused on bacterial studies because we have more information. But as he mentioned, there is much more than that. And now there is a lot of interest in virome, parasitome, archaeome, and so on and so forth. I put this artificially, separated, but I will show you in a moment an example that particularly the last three are highly interconnected. So if you lose antigen trafficking control, you put your microbiome dysbiosis and vice versa. If you have dysbiosis, you increase gut permeability. If you increase antigen trafficking, you make your immune system to generate chronic inflammation and vice versa. Chronic inflammation can increase your permeability. You got the point. But probably the most intriguing, where the money is for the future of personalized medicine and possibly primary prevention, particularly we are in the setting of pediatrics, so we really care about primary prevention, is the appreciation that the epigenetic role of the microbiome to decide if, when, why, and how genes are put in motion start their march from genetic predisposition to clinical outcome. If we got that, then we really have target for personalized intervention and primary prevention. And again, this is another slide that we put together a long time ago, and then it was changed and so on and so forth. But anyhow, to exemplify what I mean for march at the mucosal level, again, here is the GI tract, but you can put any kind of interface, the airways, the genitourinary tract, the skin, under very specific circumstances. But the bottom line is, if you follow this slide from left to the right, you see how you move from health to disease. One is when everything works the way it's supposed to. You know, the microbiome is in neobiosis, so there is good antigen trafficking that is tightly controlled. You know, the immune system is not instigated by antigen that comes in, and you reach what we call mucosal tolerance, homeostasis, that require different players there, specific immune cells, specific cytokines, and so on and so forth. The problem arises when you go from 1 to 2, in which you start to have loss of control of antigen trafficking, mainly in the space in between cells, what we call the paracellular pathway that in the past was considered to be completely sealed. Now we know that it's extremely dynamic, controlled by very complex structure that we call tight junctions. And if this antigen traffic goes out of control, there is this flux of non-self antigens, including endotoxin from the microbiome, that eventually are seen by the immune system and the lamina propria as enemies, and the immune system does what it's done for a living, fight. And you fight using, you know, modulators of inflammation, including specific cytokines like interferon gamma and TNF-alpha that per se increase gut permeability, and now you are in this vicious circle. So antigen trafficking, pro-inflammatory cytokine that increase even more antigen trafficking until you eventually go from 3 to 4. So you break tolerance, mucosal homeostasis, and you develop a disease. Which one depends who you are genetically speaking. In other words, what is on the other side of this barrier? If you are predisposed for, I don't know, functional disorder, you develop IBS, Th1, immune response, cancer, Th2, allergies, Th17, autoimmunity, and so on and so forth. One information that we didn't have for many, many years was how you move from 1 to 2. What is the pathway or the pathways that, you know, make you to lose the barrier function and eventually have this increase of gut permeability? And that's our contribution to the field. Now, almost 20 years ago, the discovery of this molecule, this pathway, the zonin pathway, that so far is the one that have been linked to a variety of these chronic inflammatory diseases. Let's see, I thought I had another slide here, but anyhow, that we'll see in a moment. But, you know, again, to exemplify what I was telling you, how a pathway like this is highly interconnected to the immune system and the dysbiosis, I want to give you a very quick example of this transgenic mouse that we use now for many of our studies. This is a zonulin transgenic mouse, produced too much zonulin compared to the wild type, and indeed, the permeability of this mouse is increased in the GI tract, both in male and females. And when you look at the microbiome, compared to the wild type, it's totally segregated. So you can see there is totally different composition. And if you ask what makes the difference, it looks like that this transgenic mouse lose protective elements of the microbiome, like Akkermansia, and has an exaggerated representation of pro-inflammatory components on the microbiome like Rickenella. And if you look at the immune system of these animals, you have the same story. I don't have time to go through much in details, but in this zonin transgenic mice, immune cell lineage that put brake on inflammation are, you know, underrepresented, where there is an overrepresentation of immune cells that are pro-inflammatory. Again, all to say, you touch one, you touch the rest, the other two elements. Here is a list, not even complete list, of the more than 800 papers in the literature growing on zonulin, including the two that I highlighted in red, the ones that we care about, you know, the Symposium, IBD, and celiac disease. Now, this is a biomarker of loss of barrier function that is used by many folks, and again, mechanistically linked to the break of mucosal tolerance that eventually leads to a chronic inflammatory disease. What are the two triggers of, the release of zonulin and upregulation of this pathway? Two that we really care about this symposium. One is gliadin, one of the components of gluten. For those that don't know, you heard from Hillary that the gluten-free diet is the cornerstone of the way that people with celiac disease are treated. This is a strange set of proteins. This is alpha-gliadin. Because of the richness in protein and glutamine cannot be completely digested by our digestive enzymes. There are undigested peptides hanging around there, and they have biological activities. The two that you see in blue are the ones that interact with a specific receptor, CXCR3 receptor, releasing zonulin and start, these issues. The second and much stronger stimulus to release zonulin and upregulate this pathway is dysbiosis. And as I told you, there is a mutual influence of each other. So dysbiosis can release zonulin, therefore increase gut permeability. And that brings me to go a little bit more in details on the microbiome. Of course, I start this chat by saying, listen, we were at war against microbes because it was the main reason why we got sick and died. Little we knew that pathogens are the exception. The rules are this wealth of microbes. They try very hard, despite all the the mistake that we make, to live in a friendly relationship with us. What they got out of this relationship is pretty obvious. They got a biological niche and they got food. Why we should engage in this symbiotic relationship? What we got out of that? A lot. We can scavenge extra energy from food that are not capable by our digestive system, like using fibers. They regulate our metabolism and therefore our clinical destiny big time. They produce a sense of vitamins that we cannot, have a good access through diet. As you see in a moment, they regulate our immune system, and this is extremely important. And last and not least, they protect against our pathogens. After all, penicillin is nothing that a molecule produced by a microbe to kill another microbe. But all this implies that we are in a friendly, symbiotic relationship that is very unique. There is a lot of effort to find, quote unquote, the normal microbiome. There is no such a thing. If we are, as we are, all genetically different, even monozygotic twins, epigenetically different, the microbiome to fit that genetic needs is totally different from each other. So there is no such thing as the healthy microbiome, like there is no such thing, what is the normal length of air that we should have? It's so personalized. What is not negotiable is the outcome of this. That is common to all of us. If we have a blood pressure within a certain range or glucose level a certain range, this is thanks to the metabolic pathways that are synchronized between these two worlds that they need to work, in cooperation. Can ski a squat here, doesn't matter. But the point that I'm trying to make is no matter what kind of environmental trigger you want to throw on the table, mom's prenatal, lifestyle, does she live in a rural urban area? Does she smoke? Drinks? Is she exposed to pollution? Perinatal. Am I born at full term or preterm? C-section or vaginal delivery? Did I spend time in the NICU or not? Breastfed or bottle-fed? Or postnatal? Infections, stress, anything in the lifestyle has been proved by gazillions of papers to affect the composition and the function of the microbiome. And with that, you may affect antigen trafficking, and you start this famous march from genetic predisposition to clinical outcome. Why, if I embrace a worsened lifestyle, I increase my risk to develop these chronic inflammatory diseases? Simply because we derailed from the plan of evolution. We were planned to be born by vaginal delivery, to be fed food and not junk. Infection, yes, but very little infections because that's the way that we died 2 million years ago. And if you survive, you're protected. Of course, there were not antibiotics at that time. And that led the microbiome to mature the right way, to be in balance. and should not come as a surprise, that is the microbiome at a specific time of our life, this famous 1,000 days from conception to 2 years of age, that will program the immune system to decide where to put the bar for inflammation. Remember, we were sick and died only for this single cause. Life expectation 14 years, 2 million years ago. Either you die of infection or a dinosaur will eat you. Will you not have the time to develop cardiovascular diseases or cancer? And inflammation is nothing else that creates a very hostile environment for any microbe to grow. It's too hot. There are chemicals like cytokines that kill me, cells like immune cells that will eat me. The tissue will die. But the entire organism will survive. And that's the reason why it's so important to program the immune system to unleash this tremendous, powerful tool only when we are under major attack, and to turn it off when the enemy is being defeated. We're not capable to do this anymore because now we're on the right side of this slide. We're born by C-section. That is totally welcome when it's medically indicated, but not to organize the OBGYN vacations. We feed junk. We feed junk to our kids. Use and abuse of antibiotics. Mea culpa, pediatricians have been responsible for this as well. And of course, infections are redundant and multiple, particularly in the first 2 years of life. And this leads to dysbiosis, and therefore the wrong programming of the immune system that does inflammation even when it's not needed. It is a question of time that this promiscuous inflammation and specific genetic background will lead to break tolerance and develop this problem. So that's where we stand with celiac disease. That's where we stand with IBD. If we got this right, now we have target for possibly change this situation. But the few other information that are important to appreciate, particularly in terms of primary prevention, is when you engraft, in a birth, of course, but now there are evidence that suggests that even in prenatal era, we have the first encounter with our microbiome, even in conception. So there is a father component. Some people that believe. This is not solid science yet, but there's a lot of theories that, there are people that, believe strongly about a placenta microbiome, other people that do not, and the blood microbiome and so on and so forth. The bottom line is, particularly in the last trimester of pregnancy, if indeed, looks like there is the initial encounter with our microbiome, mom lifestyle counts. But also when she will pass, what is coming. If you look at the microbiome from birth, it's completely chaotic. There are some microbes that get in, then they leave. Then there is another shift when baby food is introduced. And then finally, everything seems to quiet down around 2 years of age when the microbiome reaches, the adult maturation. Chaos? Dating. We're searching each other. The microbes are checking us and say, you are a good biological niche for me. I will stay. I will settle here. Or I don't see compatibility. I better get out of here and find somebody else. And we do the same. We say, you will be useful for me. You can stay. Or you will not. You will be detrimental. You got to go. And imagine the impact on this very delicate mechanism and path of choosing each other, what kind of consequences it can have over time. So that's the reason why those 5,000 days of life are instrumental. I'm going to use my last part of the discussion to go into the details. With that premise, with that kind of background, what we can do going forward, and I'm using celiac disease as an example. Now, the entire world of the microbiome is at the tremendous crossroad. Most of the literature, including ours, has been very descriptive. You take, a certain number of people with a disease, let's say celiac disease or IBD, a certain numbers that will not have the disease. You look at the microbiome, it's different, and then you put the loop of fate conclusion, you got the disease because of the microbiome. It can be the other way around. Simply because I have an inflammatory gut, that can change the microbiome. That so it can be the consequence and not the cause. The only way to mechanistically link shift of the microbiome to disease onset is longitudinal studies. Take folks possibly at risk and follow over time so that you understand the march that we discussed before, at the onset and after the onset of the microbiome, and then and only then, compared to the ones that develop the disease versus the ones that do not, you can make a mechanistic link there. And this is what we've been doing with this study. It's called CDGEM. It's an acronym for Celiac Disease Genome Environmental Microbiome Development Studies. This was started 10 years ago when the microbiome studies were still in its infancy, very expensive. 16S was the only way to do that, and so on and so forth. You know, in tune with what the center here for celiac disease at IBD is doing, all this kind of exercise would be useless without a biobank. You can be as smart as you want, clinically or, you know, scientifically, but you don't have the tools if you don't have a good biobank of samples, but if you allow me, even more important, a biobank of metadata. Not having clinical information, it's like that you want to understand the Egyptian hieroglyphic without having key to understand what are symbols they mean. Just to give you an example, we have a biobank with 125,000 samples, 170 data points, and so on and so forth. And like also the other, chronic inflammatory diseases, even, celiac disease is increasing over time, doubling every 15 years. So no change in genomics. Of course, the grains did not change, so the microbiome seems to play here. And I'll give you a quick example of analysis, longitudinal analysis of microbiome done in this cohort that showed, interestingly, that those kids, these are kids at risk because somebody in the family has celiac disease, that will develop celiac disease, will have an increase in abundance of microbacteria and metabolites that were previously linked to pro-inflammatory pathways, and have a deprioperation of protective elements, and the opposite of control. And if you ask the question, and again, we focus on bacteria not because we don't like parasites or virus, but because the libraries are very extensively done in bacteria, not yet for the others. And you ask, what makes the difference? These 5 strains, only these 5 strains, 100% of the time, segregated cases and controls. These are strains that are lost for kids that develop celiac disease that were still there in the one that, despite to be at risk, they did not. And these are all probiotic strains that have been described in the past. Now, contrary to what we reported before, these are all cultivable strains. You sequence whole genome sequencing, and you see the difference, 2% of genome different from the reference strains, and you ask, what these genes are mutated they do for living? They interact with those controlling what? 3 domains. Barrier function, so antigen trafficking, immune response, and cell turnover. So stuff that you will anticipate in a situation like this. And again, using gut organoids of these kids that we do in 3D, as Michael was referring, but also in 2D, we eventually put gluten there because we have the luxury to know what is the instigated inflammation that we do not have for IBD. And we look at what happened in the presence of the gluten with or without this protective strain. And long story short, if you put gluten, guess what? You increase gut permeability in a zone-independent way. You have pro-inflammatory cytokines that are speeded up, and there is increased cell death. So apoptosis, all intercepted by these microbes. And how they do that? Epigenetically. Once again, what these microbes they do on the epithelium is to touch genes that control epithelial cell turnover, barrier function, and innate adaptive immune response in a way that say, if you see gluten, do not do anything. Just stay put. That's fine. Don't get upset. We will take care of it. So that's the epigenetic capability to protect. So this is, in my humble opinion, this is a paper that will be published soon, is the first example in which we see mechanistically how specific strain in the microbiome can eventually protect against an inflammatory process. And if you look at the evolution of, for example, zonin as a marker of permeability, once again, compared to the control, the blue line, there is no change over time.
the ones that are destined to develop celiac disease, they have a rapid increase of zonin over time that precedes months the onset of the disease. And among all the environmental factors, the most impactful is how many antibiotics these kids they take. The more antibiotics, the more that curve becomes steeper. And that means that, again, it's a big, elements. And also, there is a possibility to predict who develops celiac disease based on, the biomarker of loss of tolerance to gluten with deamidated gliadin. Bottom line, years before we know what is the dysbiosis, and a year and a half before we see this epigenetic rearrangement, a year before we see zonin go up, 6-9 months before we see the break of tolerance to gluten, and then the onset of disease. All target for possible intervention. And I'm going to finish up by saying this is going to be the future. Machine learning, big metadata that you put in there together with microbiome and so on and so forth to try to really come up with model, including metadata with wearable devices and so on and so forth. But Michael Snyder will talk a little bit more about this. And of course, the last thing that I want to say in the last 2 minutes that are left is what would be the low-hanging fruit to manipulate this, to really revert to what we have seen. We're born once, we take antibiotics 2-3 times a year, but we eat 3-4 times a day. Nutrition is by far the most impactful factor that changes the microbiome composition and function. And now, 2023, the main reason why you are sick and die is not infections anymore, is nutrition. Look at this map. The darker, the more impactful in nutrition. And all the stuff that goes wrong, that increases your risk, is all stuff that has to do with the microbiome. Diet poor of fibers in fruit and vegetable, nuts, tubers, and so on and so forth. These are the stuff that the microbiome thrive to, and they don't have food anymore. And this is the key elements of the Mediterranean diet. That is not just a diet, of course, it's a lifestyle. Meaning that, you can eat as good as you go, but you spend 10 hours on the couch, that doesn't help. So the bottom of the pie pyramid is physical activity and all the other yards. And the future, as we talk about with Michael before, precision N=1 precision, medicine is going to be precision nutrition. The diet needs to be tailored on specific people, and there is no one size fits all. Genetic, epigenetic, microbiome, metabolomic, all integrated will be the future. Remember, final message, we as pediatricians can really integrate the effort of the adults because the game starts there, and you set the destiny of these kids in the first 1,000 days of life. So a good lifestyle at that point can be really impactful. So to finish up these food for thoughts, not pounding in tender, the established possible role of gut microbiome in the pathogenesis of chronic inflammatory disease, you need to have these longitudinal studies, needs to be interpreted, the microbiome in the context of other multi-omics, otherwise you would not make sense for personalized intervention. And you see that there is a highly predictable, 93%, we can predict now who down the road having a specific microbiome profile would develop celiac disease. And all the intervention, probably nutrition will be the easy way, not the easiest, the easy way compared to other interventions to manipulate our destiny. If you are interested, just take note that next year, June 22, 25, there's going to be the 10th International Human Microbiome Consortium. The team is One Health. Rome is a beautiful place to visit if you've never been there. So that's, just put the plug in there. If you're really interested to expand what you heard today, this is an opportunity. I'm going to finish again to thank for the kind invitation, to sit on the board of the center, all the funding agency. This is a very short list of colleagues that, have been involved. There are many more. But most important, that picture there, these are our CityGem kids. These are kids that when they got to the first year graduation of participating, the society, they come to the lab, they got the lab coat ceremony, they got their own lab coats. So it's something that you guys should really consider with the logo and everything. And they are so excited. The thing that they are excited the most is to look at the freezer, they poop frozen in there. Thank you so much.
Okay, so thanks for that really exciting and provocative talk, and we'll take some questions.
for your wonderful talk. So I have a question about the very young children who get celiac disease, like under 5 years of age. They may not have had enough time to be on something like Mediterranean diet, etc. Do you think there is a role for patients who are genetically predisposed, like a strong family history, to do some of the, say, probiotic or diet interventions very early in life?
So, as you know, celiac disease was considered historically a pediatric condition because that's where it starts, in childhood. The study that we did, in CDGEM shows that 80% of who has to lose tolerance, it does really early, in the first 5 years of life. And interestingly enough, we have two cohorts to establish the environmental component versus genetic, as you were mentioning. One is in the United States, the other one is in Italy. We didn't see too much genetic difference between these two cohorts, but in Italy, the acceleration of losing tolerance is 3 times faster. So there are 3 times more kids that develop celiac disease very early compared to the United States, but then eventually they catch up later on. Meaning that the environment, the way that we feed our kids, that makes big difference to lose tolerance and start that march from the predisposition to clinical outcome.
Thank you so much for this talk. That was great. In the data you have shown us, you have kind of suggested that the increase in the zonunin and the cytokine was what preceded the development of celiac disease in infant at risk, what kind of alluding to the fact that gut permeability is what starts causing the disease. There's similar kind of models that we have seen in other type of disease, but more chronic, like in chronic HIV, where you would have the gut permeability that will eventually lead to some of these complications. I was wondering, what was your thought on why in these patients the destruction cannot be repaired on its own? Like when it's chronic disease, you know that you have, the destruction has been so severe to the point where you cannot regenerate the epithelia, but versus in here, what kind of continuous insult can be contributing to this persistent damage?
So I'm going to respond in a very provocative way that would not be accepted, particularly by my colleagues, immunologists. The general belief nowadays is that if you break tolerance and you develop an autoimmune disease or chronic inflammation, that's a one way. You can't repair that. Now, just think about evolutionarily speaking. What would be the advantage for me to have the genes that put me at risk for celiac disease, IBD, type 1 diabetes? I have no advantage. And how you explain epidemiologically that there is no genetic, elimination of that mutation? Actually, there is an acceleration. The only logical answer is that this is not a one-way street. It's just the continued exposure of the immune system to something that instigates the immune system to fight the problem. If you repair that, probably you will be able to, a matter of fact, celiac disease is an example. There have been, inhibitors of the zonunin in the clinical trials in which you keep gluten in there, so the instigator, but you give zonunin, and yet you can achieve the same goal. And we've seen in animal models and so on and so forth. This is a long way to answer your question. Easy to say, very difficult to implement. Because remember, destruction of this homeostasis is the consequence of dysbiosis. And dysbiosis is the consequence of the way that we embrace the lifestyle. We are not respecting and taking care of our microbiome. If we will go back, not to live in a cave, because it's impossible, that would be inconceivable, but to respect our microbiome, again, nutrition would be the easy way to do that. Chance exists that we will mitigate that loss of barrier and therefore the continued instigation of the immune system to generate chronic inflammation in HIV, in celiac disease, in IBD, and so on and so forth. By the way, IBD first line of intervention now for remission is not steroids anymore, it's elemental diet, and that speaks volume. Of course, you can't have this sustainable over time. Guess what's happening to the microbiome just shifting on elemental diet.
Any other questions? Oh, yeah. Okay. All three of you in one order after another.
Thank you, Alessio. This was fantastic. This kind of goes along the lines of what Alka was asking, and I apologize if I just didn't quite catch the answer. But is there any way, I know we've talked about the microbiome and its impact on going on to develop celiac disease, and the impact of certain aspects of the diet to promote a positive microbiome that could be protective. And so is there any way in patients that are high risk for developing celiac disease because they have a family history of flooding them with very specific probiotics to overcome that risk and allow them to not go down the path maybe they were genetically predisposed for?
Yeah, I mean, those 5 strains are all patented, and now a clinical trial is starting for these kids that they have genetic predisposition, that they lose that, components, because they have a very, very high chance, 93% within a year, to develop celiac disease. We'll have an answer when these trials will be finished. But again, this is the Band-Aid. I believe that the future, I don't know if Michael Snyder is here yet or not, but it's what he's going to tell us, this major metadata and multi-omic analysis that will really, what I see in the future, in other words, is something that comes to your office and you have the whole genome sequence, because we all will have a birth, bring the stools and the urine, you put in the machine in the back of your office, you do my physical exam, at the end you finish, you put that in this mega, database that compare me with millions of other people. Say, Alessio, in 5 years, you have a risk to develop Alzheimer. You got to change your diet or the microbiome left or right. And that's going to be the heart and soul of personalized medicine N equal 1. That's what I think that's going to be the future.
This is going to be a much more reductionist question. So I love Michael's talk and yours, the use of organoids as a human model to study disease mechanisms. But I was interested, does the model organoids you're using have immune cells? And if not, how is gluten causing problems in the epithelium without an immune system?
So 2 answers. One, we do in co-culture with macrophages and other specific immune cells that we're, interested. But in a very provocative way, I will answer, who cares? Gluten, in order to instigate the immune cells, has to break tolerance at the mucosal level. The game is at the epithelial level. If you do not have gluten coming through, the immune cells will not see it. And the enterocytes, they have immune response capability. It's all innate immune response. First thing that happened on an organoid, just no culture, just epithelial, is speeding up IL-8. And we know that the first thing that happened when you're exposed to gluten is this inflection of neutrophils coming in the lamina propria that will start the insult that leaks, TTG out of the cells, and then, when gluten comes through the deamination and so on and so forth. So, in other words, the epithelium has been always overlooked, I believe. And we need to change our mindset, because all the inflammatory processes that we go after with anti-inflammatory drugs, you treat the consequence and not the cause. It's like they have pneumonia and you take Tylenol. Of course, fever goes away, but the reason why you have fever is still there. The ambition that we need to really step up with IBD and celiac disease is that why I'm generating inflammation and going after that mechanism.
Is the innate response to gluten specific to patients with celiac disease, or is that a more global thing that they're just not?
It's more global, but the consequences are very narrow to the people that once gluten comes on the other side, they are not capable to manage that correctly. So the innate gluten is managed by the epithelium like a component of microbes, pretty much. Activate the same toll-like receptor 4, specific response, other part of the recognition receptor, NF kappa B activation, another killer cell. So in other words, it really mimics what you see when we have to deal with the microbes. And again, it's the epithelium to decide if this is an okay situation and stay in homeostasis or not. I'm not an expert in IBD, but the vast majority of you guys believe that IBD is the consequence of not managing that right. So we all have healthy inflammation in the gut. It goes out of control if you're genetically predisposed to not manage that encounter microbes in a healthy way and generate chronic inflammation. All right, yeah, time is over. Thank you.
Okay. Thank you very much, and thank you all for your questions. Let's see. So we'll get back here at noon, and we have a break until then.
Yeah. Okay, perfect. This thing maybe? Or? What? Okay, yeah. Uh-huh. I think so. Uh-huh. Okay, yeah. Yeah, 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 yeah. Hello, everyone. Welcome back. I'm excited to welcome you to this next session here, where we have another two great speakers. The first is a keynote address as well. First, I'd like to welcome Dr. Maria Oliva Hemker. She's the keynote speaker of the session. If you were at NASPGHAN last month, you may have learned a lot about her. She was the recipient of the Davidson Award. Dr. Oliva Hemker is the Sturmer Family Professor of Pediatric Inflammatory Bowel Disease and Professor of Pediatrics at the Johns Hopkins University School of Medicine. She launched the Pediatric IBD Center at Johns Hopkins, and Dr. Oliva Hemker has been the chief of the Division of Pediatric Gastroenterology, Hepatology, and Nutrition at the Johns Hopkins Children's Center for the past 15 years, and she is the Vice Dean of the Faculty of the Johns Hopkins University School of Medicine. She's led a variety of initiatives and large projects in IBD and in the field of pediatric gastroenterology, including a focus of C. difficile and IBD. I'm very excited today to hear her keynote titled C. difficile and Inflammatory Bowel Disease. Please join me in welcoming Dr. Maria Oliva Hemker.
Okay. Thank you very much, Ruben, and it's certainly a pleasure for me to have the opportunity to talk to you folks today. So, I am a clinician and a clinical researcher, been taking care of kids with inflammatory bowel disease for, now I can say the word, decades. And so what I want to talk to you today, and hopefully engage some of you who have interest in infectious diseases and inflammatory diseases, I want you to talk about the impact of C. difficile on our patients with inflammatory bowel disease. And I'm not going to speak specifically on IBD. I know many of you are aware of inflammatory bowel disease, the umbrella term that we use for Crohn's disease and for ulcerative colitis. And as you've heard mentioned today, these are chronic inflammatory conditions. We don't really know what the true cause is, but you need that genetic risk factor. You need environmental stimuli, and then you need a dysregulated immune system. And right now, the treatment is primarily immunosuppressive conditions. These are my disclosures that are pertinent for this talk. All right, so I'm going to start with a story. Why do I have a little bit more than a passing interest in C. diff and IBD? Well, when I was in my early career of faculty, a mom brought her 2.5, 3-year-old son to see me. And he had been suffering from recurrent bouts of C. difficile. And she came to me because she had heard somewhere that I was a probiotic-friendly doctor. And I'm not quite sure what that meant, but I did use some probiotics at the time. And so she found, and she came to me. And as she was giving me the history, she took out these papers and she put them on her lap. And I could only see them kind of, on the opposite side, and I saw the word probiotic. And the mom was going on and on about probiotic therapy, and then she mentioned, oh, and I'm getting a new blender out of this. And I thought, wait a second, what do you mean you're getting a new blender out of this? And she was like, oh yes, because our old blender is going to be used to mix up my husband's stool, and then we won't be able to use it anymore, and that will be the probiotic therapy that we're going to give to our 3-year-old, but if you help us, maybe we won't need to do that. And when I said, well, let me have those papers, and I went, oh, that probiotic, you mean he's talking about stool? This was in the 1990s, folks. And I said, well, give me a second. Let's actually treat your child with another vancomycin taper. Let me give him some additional probiotics, the regular kind, not the stool kind, and let's see what happens. And thankfully, her son did get better. I followed him for a while. We were able to stop the probiotics, and he never had, to my knowledge, another episode of recurrent C. diff. Now, that mom was a PhD, and she was at Penn State at the time, and she became intrigued with probiotics, so she ended up writing a book called Bacteria for Breakfast that you can still get on Amazon today. And I did connect with her about a year ago. She's now a professor at East Tennessee State, and her son just graduated from college, and he never had another episode of C. diff. So before they left clinic that day, I took the papers and I made a copy of them, and I filed them under kind of the strange things people do kind of category, and then I forgot about them for a while. But that was my first kind of thought about C. diff, but also about stool therapy, and I'm going to get back to that in a little bit. All right, so what are the objectives of my talk? I'm going to tell you a little bit about the epidemiology and impact of C. difficile infection in IBD. I want you to understand the difficulties in diagnosing C. diff infection in our IBD patients and review the recommended strategies to treat C. diff. So let's start with the organism. People may have remembered it to be called Clostridium difficile. It's now Clostridioides difficile. The name was changed in 2016 when the organism was reclassified. It's a gram-positive, obligate anaerobic bacteria, and it is the toxins that are secreted by the toxigenic C. diff strains that damage the intestinal epithelium. So it can be easily spread. It can persist in the environment. Those of us that work in the hospital really have heard about this for quite a bit of time. And it is because the fact that the organism can become spores, the spore formation that makes it so difficult to sometimes treat and get out of the environment. A little bit of history here. This was actually discovered by Hall and O'Toole in 1935. And these investigators started culturing the stool of healthy newborn infants for the first 10 days of life. And out of that culture, they ended up discovering this unusual gram-positive rod that had not been classified before, and it took them an incredible amount of time to really get these organisms to grow. And that petri dish is the 48-hour growth when they were finally able to find the right media. And because it had been so difficult to grow, they named this Bacillus difficilis. So that's how it got its name. Now, interestingly, they actually injected this organism into guinea pigs and rabbits, and to their total surprise, the animals went totally apoplectic. They became very ill. They went into convulsions. Many of them died, and these investigators thought this is just so unusual that an organism that's residing in a healthy newborn infant can create so much damage in these animals. But then, Bacillus difficilis and then C. diff basically went into the background for about 4 decades, and it didn't become well known again until 1978, when it was identified as the major cause of pseudomembranous colitis. And that was an article by John Bartlett and colleagues, and Dr. Bartlett actually ended up going to Hopkins, stayed there for decades. He was the division director of infectious diseases and became known actually even more for his work in HIV/AIDS. So C. diff infection can have a very wide variety of presentations, from asymptomatic carriage to mild self-limiting diarrhea, to moderate diarrhea, to the more severe forms that include pseudomembranous colitis. There's a photo there of what the colon would look like, bowel perforation and septic shock. And thankfully, in pediatrics, we don't see many patients at that severe end of the spectrum. The interesting thing about C. diff and how we treat it is that once you get an infection, you're at risk for developing a recurrence. And I'm showing you there the recurrence rates in the general population, maybe 15 to 35% after having one episode of C. diff. And the more episodes you have, the greater the recurrence rate. And if you look at these numbers and think about patients with inflammatory bowel disease, those patients have an even higher recurrence rate than even the general population. So this is what makes C. diff kind of a difficult infection to be dealing with. The CDC has called C. diff kind of an urgent infection, an urgent antibiotic-associated infection, and it's the most common cause of antibiotic-associated diarrhea, most common hospital-acquired infection, with an estimated annual burden of cases of nearly about 500,000, with more than 30,000 deaths per year. And again, most of this is in the adult population. Thankfully, there has been a decrease in the burden of disease since about 2011, and that has been primarily due to a decline in the healthcare-associated infections that you see there in the bottom graphs. However, the community-associated infections remain unchanged, and they currently contribute to about 50% of the current CDI burden. And as you can imagine, the healthcare costs for something like this are quite high, at least in about the $5 billion range. The impact of C. diff infection in children is significant as well, although children certainly don't get C. diff as often as older adults. But it's estimated to be about 20,000 children per year, with about 30% of children having recurrence of an infection within 60 days. And in children, and children with IBD, it's the same thing. The vast majority of cases are acquired in the community and not necessarily in the hospital setting. So what happens with C. diff infection and IBD? Well, here is a list of varying clinical studies that looked at both adults and pediatric patients, and you see there the time periods of the sampling. It was a whole variety of inflammatory bowel disease patients. Some were in disease flares, some were not. But in general, when you look at the percent of IBD patients with C. diff positive stools, what you see here, compared to the baseline of maybe 1 to 3% in the general population, is that patients with IBD typically have more C. diff positive stools, anywhere from about 6% to a high of 47% there. So IBD patients tend to have more C. diff positivity than the general population. So about 10 years ago, I was lucky enough to have a very smart, pediatric GI fellow who came to join us, and she was interested in doing microbiome work and in some IBD work. Her name is Suchi Hurigan, and we were talking, got together, and decided that Suchi would go work in the lab of a person who has become a very wonderful collaborator of ours, Cynthia Sears, who's a professor of infectious diseases. And as Suchi went into the lab, you know, we already were aware that there was increased C. diff infection in IBD patients and peds and adults, but we decided that we wanted to have a kind of a better epidemiologic study, and we partnered with Susie Hutflis, who is an epidemiologist at Hopkins, and we decided to look at our own state of Maryland using a hospital discharge database of 50 Maryland hospitals. And what did this show us? Well, it showed, when we looked at a 20-year period from 1993 to 2012, that cases in hospitalized patients of C. diff were increasing in children and in adults, and you see that in the dotted lines, with the pediatric patients without IBD being the dark dotted lines, and the lighter lines are adult patients. But this also allowed us to compare and look at peds and adult IBD, and those are the solid lines. And as you can see, over time, the rates of hospitalized patients with inflammatory bowel disease and CDI were increasing. In fact, over that period, if you look at the bottom section of the slide here, you will see that children who are hospitalized with inflammatory bowel disease had more than 12 times the chance of being also infected or having C. diff positive stools than children without IBD, and then in the adult population, it was 4 times more in adults with inflammatory bowel disease. And then as we were looking a little bit more deeply into this, we knew that there had been some adult studies that had been done in Ireland by Clayton and colleagues who had looked at asymptomatic adults with inflammatory bowel disease and had done anaerobic stool cultures. And they had discovered that in adults with IBD who were asymptomatic, approximately 8% of them had C. diff, toxigenic C. diff in their stools, compared to 1%, which is what you would expect for the general population. So Suchi Horrigan decided that this was something that she wanted to look at, and we began enrolling children from our IBD clinic and children from other parts of our regular general GI clinic that did not end up with any diagnosis that led to an inflammatory underlying condition, and we used those children as controls. And doing kind of the gold standard evaluation for C. diff, which is culturing the organisms anaerobically and then doing a cytotoxic assay, so the most sensitive but the gold standard way of looking for C. diff, what we found was that actually in our asymptomatic children with inflammatory bowel disease, 17% of them had C. diff in their stool compared to 3% in our control population. And as we looked at the various strains that were identified in the children that had IBD, they actually had different strains over time, because the beauty of this particular study was that we did end up acquiring stool samples from a good portion of the patients over a period of time. So children and adults with inflammatory bowel disease, even without having disease activity, being asymptomatic, were carrying C. diff in their stool. So what are the adverse complications of having a C. diff infection if you are a patient with inflammatory bowel disease? This is where the problem lies, and from a clinical perspective, makes these patients very difficult for us to treat. So it's been found that if you have the concomitant conditions, CDI and IBD, that there is a greater chance to having flares of your inflammatory bowel disease, that you are more likely to require that your IBD therapy be escalated, and because you're more likely to fail the current therapies. Higher surgical rates, and in this case, it's often been described as either intestinal, but primarily colectomy rates are higher in patients that have inflammatory bowel disease and CDI, and it's been pretty well established in pediatrics and in adults. Longer hospital stays in the range of about 2 to 3 days on average. Increased emergency room visits. Again, I already mentioned that if you have IBD, you have a greater risk of having more frequent recurrences, and a higher mortality than if you just have IBD alone. In fact, having C. diff increases mortality rate by 4-fold, and that's primarily, again, in adult patients. And as you can imagine, increased healthcare costs. Now, in the general population, the biggest kind of risk factors are associated with the pathogenesis of C. diff infection is antibiotic use and hospitalizations. But the IBD population looks a little bit different in this case, and I'm showing you here differences in patients with and without inflammatory bowel disease. So the patients with IBD who have C. diff infections tend to be younger. Their antibiotic exposure occurs less than in the general population. They're acquiring their IBD, they're acquiring their C. diff infection typically from the community rather than the hospital setting. As I already mentioned, asymptomatic colonization is more common. The recurrence rates are higher. And if you look at the intestinal environment, it has already been raised by other speakers today. In IBD, you have a persistent dysbiosis due to the underlying disease condition, whereas in patients that don't have IBD, although they have been exposed to antibiotics, it's felt that those infrequent exposures has less of an overall impact in their microbiota compared to patients with inflammatory bowel disease. So I know that there are a number of people in this room that can speak very deeply about the intestinal microbiome changes in patients with inflammatory bowel disease. So I'm going to talk about this, though, at a higher level. Multiple studies have shown that compared with healthy subjects, the microbiota of patients with inflammatory bowel disease have reduced overall microbiota diversity, a reduction in bacteria that produce anti-inflammatory cytokines, reduction in bacteria that produce short-chain fatty acids, which are a very important fuel, especially for our colon, and have a reduction in secondary bile acids. And that beautiful Circos plot that you see there on your right just highlights some of the bacteria that have been associated in the pathogenesis of Crohn's disease and ulcerative colitis in terms of their general abundance. So the red ribbons are showing you bacteria that are felt to be more pathogenic in these particular environments with higher levels, and they include, for example, as you see there, E. coli and Ruminococcus species. And then the blue ribbons are representative of what people would term kind of healthier bacteria, and those are bacteria that are found in lower abundance in Crohn's disease and ulcerative colitis, and they include bacteria, for example, like F. prausnitzii and Roseburia intestinalis, and those are two bacteria that actually tend to produce a lot of short-chain fatty acids, again, good for colonic health, but in these particular cases in inflammatory bowel disease, they're found in lower abundance. When people have looked at IBD and C. diff infection, again, similarly low microbial diversity has been shown. And Leanne Chen, who was an adult GI fellow at Hopkins, and then she went off to Rutgers and had been working in Martin Blazer's lab, continued to collaborate with us, and she did a study that showed that children with IBD who were asymptomatic and colonized, but they had C. diff, also had reduced bacterial diversity and altered bacterial composition. So the diagnostic criteria for C. diff infection appears to be pretty straightforward when you kind of look at it this way. You need to have diarrhea. You may have radiographic evidence of ileus or toxic megacolon. The X-ray there is of an adult with toxic megacolon, dramatically dilated colon. You may have visual or histopathologic findings such as pseudomembranous colitis, and you may have a positive stool test for toxigenic C. diff or a positive stool test for the toxins. It seems intuitively pretty easy, but it's not so much. So whether you have inflammatory bowel disease or not, the methods to diagnose C. diff infection are the same. I briefly already mentioned the gold standards that we were doing in our research studies, and they include cell culture of C. difficile, and then in order to know whether you have a toxin producing a strain, you need to do a cytotoxic assay. But what's the problem with these? They are time consuming. You need personnel that are trained. The turnaround time is much longer than one would want to have in order to have a clinical answer as to whether my patient right now has a toxin producing C. diff infection. So rapid diagnostic tests came out in the 2000s and beyond, and these include enzyme immunoassays, as well as nucleic acid amplification testing, or the PCR testing, which is now how routinely we're making these diagnoses. The problem with these PCR-type tests, of course, is that they are exceedingly sensitive, and they're testing, in many cases, for the genes that produce the toxin and not necessarily the toxin. And so we may actually be detecting, and I know we are, colonization rates in patients rather than patients that have actually an active C. diff infection. So this is the challenge that we have in diagnosing C. diff in our patients with inflammatory bowel disease. The symptoms of C. diff infection, diarrhea, maybe blood in the stool, abdominal cramping, can be indistinguishable from the symptoms that a patient with inflammatory bowel disease may have, especially if they have colonic disease. The biochemical markers that we use, for example, the fecal calprotectin, are generally unhelpful. The high rates of C. diff colonization that I have already mentioned to you, 17% in our pediatric patients with IBD, impact our ability to diagnose true infection. And the pseudomembranes that are often described in the textbooks are not commonly found in inflammatory bowel disease. In fact, there was an adult study where only 13% of 93 hospitalized adults that underwent colonoscopy actually had pseudomembranes. And now that I've done colonoscopies on quite a number of patients with inflammatory bowel disease and CDI, I have yet to see a case of pseudomembranous colitis in one of our pediatric patients. It's very rare. And although the infectious disease societies and the hospital epidemiologists have said, well, you know, one rapid test may not be enough. Maybe everybody should be doing 2 tests using like a multi-step algorithm in order to diagnose C. diff infection. When our colleagues at Vanderbilt took a look at the multi-step algorithms that were being recommended, they found that these multi-step algorithms were not able to differentiate between symptomatic C. diff infection and asymptomatic colonization in pediatric cohorts. So now, at this point, what is the management of C. diff infection in adults with IBD? And I say adults because this is where the algorithms lie. We don't have well thought out algorithms in pediatrics, so we are basically using what our adult colleagues have recommended, which comes from both the information in the medical literature as well as expert opinion. Let me take you through some of the best practices that are now being recommended. First of all, if you have a patient with inflammatory bowel disease and they have an acute change in their symptoms, now we always need to check for C. diff infection. It's become very routine. It wasn't about 15 years ago, but now it's become routine. Second, the management and the treatment for C. diff infection in the regular population often involves starting with metronidazole as the first step antibiotic. For inflammatory bowel disease, we bypass that and go to vancomycin. That's become frontline, and if insurance approves, there's money to cover it, fidaxomicin is the other antibiotic. 3, in the past, people were afraid to alter and change the immunosuppressive, immunomodulating medications of the patients with IBD because they were concerned there's an active infection. How can I actually make my patient more, quote unquote, immunosuppressed? Now, if there's no improvement in symptoms after a few days of treating C. diff infection, escalation of immunosuppression is recommended. And then finally, I want to show you this. What happens with recurrent C. diff? Well, much to my surprise, at this point in time, maybe not so much to my surprise, but surprising myself from about 10 years ago, the now fecal microbiota transplantation has actually made it to the algorithms for the management of recurrent C. diff. And let me use these last set of slides to talk to you about this. How did FMT, fecal microbiota transplantation, come now to be part of the algorithms in the management of recurrent C. diff? Well, this is not a new thing. It actually dates back to 4th century China, when there was a Chinese physician named Ji Hong who would actually treat food poisoning and diarrheal illnesses by giving people suspensions of stool from healthy individuals. And he called it yellow soup at the time, because you really wouldn't want to say what you were giving in the soup, right? So this goes back to 4th century China, and the first mention in modern medical literature was in 1958, when 4 patients with severe pseudomembranous colitis were cured with fecal retention enemas. So go all the way now to 2013, when the pivotal randomized controlled trial showed that FMT was significantly more effective than vancomycin. 81% in the FMT patients versus 30% in the vancomycin patients. This was very dramatic, and they actually had to stop the study midway because it was felt to be unethical to continue offering patients vancomycin. And now we have a number of systemic reviews and meta-analyses that are showing that for the treatment of recurrent C. diff in adults, the cure rate at 8 weeks is 84% for a single FMT and 91% for a repeat FMT. In pediatric patients, the retrospective study of 335 patients that was led by Mary Beth Nicholson at Vanderbilt and that we all many of us were involved in, showed a rate of 81% with a single FMT and 87% with multiple FMTs. I'm going to jump now to a very recent study from this year about FMT in adults with inflammatory bowel disease and recurrent C. diff infection. This was a European study of 113 adults. 63% of them were receiving immunosuppressive medication, and 54% were not in IBD remission at the time. The success rate in IBD patients was 71%, less than in the general population, but still quite good. And during follow-up of up to 2 years, I do want to say that 5% of those patients did undergo colectomy, probably not surprising. 10% of the patients died, but this had nothing to do with the FMT. It just showed that this was probably a more fragile population with their inflammatory bowel disease. I am showing you here the largest retrospective study of C. diff infection in IBD patients and its treatment with FMT. And again, Mary Beth Nicholson was the one who led this study of 20 pediatric centers across the United States over an 8-year period. We looked at 396 patients, 148 of them had IBD, 249 did not have IBD, and most of those children had had C. diff at least for a median of 3 times previously. The success rate was 76% in our pediatric patients. Remember, it was 71% in the adult patients with IBD. That was after 1 FMT. And the good thing about this particular study was that it did not seem to impact or make the inflammatory bowel disease worse in these children having the FMT, which was something that had always been a bit of a question. So why does FMT work? Well, this is a simple schematic to say, how we were thinking about it. You have homeostasis in your GI tract, but then a C. diff infection and multiple exposures to antibiotics will lead to a persistent dysbiosis. FMT appears to disrupt that dysbiosis, allows an expansion of the commensals, and the C. diff is then cleared. When we started doing FMTs at Hopkins back in 2013, which was Suchi Horrigan's last year as a Peds GI fellow. We decided to start collecting stool from the children who were undergoing the FMTs, and we decided to collect the stools out to a 6-month time period. And what I'm showing you here is kind of a very overall view of the microbiome changes that we noted in these patients. You see the donor pool to the right, those were healthy donors, and for our patients, they were identified donors. And this represents more or less what you would expect the healthy phylum in donors, in which the majority was the firmicutes and the firmicutes and the Bacteroides in the pink and in the purple. However, pre-FMT, our patients had an exceeding amount of proteobacteria. You see that up here in the brown. After FMT and moving forward, as you can see, the phylum of the patients started to mimic that of their donors. And when we looked at the diversity pre and post FMT, we noticed that the donors who had expected great diversity in their stool, when we looked and compared with the pediatric patients, we noticed that pre-FMT, the patients that had IBD and didn't have IBD had very low diversity. At 6 months, Interestingly, the patients that did not have IBD had diversity very equivalent to that of their donors, but our IBD positive patients whose diversity had increased over the first 1 or 2 months all of a sudden had a biodiversity that was much lower and was back to the baseline. And this occurred even though all of the patients had eradicated their C. diff. So when we saw this, I thought, okay, one FMT is not going to do it, and one FMT is definitely not going to fix somebody's inflammatory bowel disease, which is the question that we were all asking ourselves when we started doing this. And then finally, let me show you here, as we noted that, that diversity can be impacted with FMT, and that the composition of the microbiota in recipients goes and tends to be similar to that of the donors. The question was, there must be something else. It can't just be the diversity alone. There must be other metabolites that are important in trying to maintain the positivity that FMT is given. And I'd mentioned early on that in inflammatory bowel disease, secondary bile acids are decreased. And the secondary bile acids are viewed as positive bile acids because they actually inhibit growth of C. diff organisms, whereas primary bile acids actually encourage growth of C. diff. And this was a study, again, it was longitudinal of bile acid composition, and what you see here that the donors in our cases were all adults. And I've already mentioned about the bacterial diversity, but you see there that the donors, when you looked at the bile acids in their feces, had high amounts of secondary bile acids, good bile acids, low amounts of primary bile acids. The children before FMT who had C. diff infection had the reverse, but then after their FMT, the children all had much higher proportion of secondary bile acids in their feces and a much lower proportion of primary bile acids. So again, showing that the fecal transplant actually impacts the metabolism in different ways. So what are the key takeaways here of this overview that I've wanted to give you with C. diff? First is that inflammatory bowel disease is associated with an increased risk of primary C. diff infection as well as recurrent C. diff infection. And this is thought to be due to GI inflammation as well as alterations in the intestinal microbiota. IBD and C. diff infection leads to worse surgical and clinical outcomes and a need to escalate inflammatory bowel disease therapies. The symptom overlap between IBD flares and CDI and our decreased ability to differentiate CDI from C. difficile colonization creates a diagnostic dilemma that we haven't yet been able to figure out. And then finally, even though antibiotics are the mainstay for initial treatment of CDI, FMT has now emerged as an effective treatment for recurrent C. difficile in patients without IBD and in patients with IBD. And I just want to acknowledge some of the collaborators that have worked with me on these studies. Like I mentioned, Cindy Sears at Hopkins. Suchi Hurigan is now at the NIH. She's actually the director of the clinical biome unit at NIAID. At George Washington, Keith Crandall and his group worked with us on metagenomic studies that I didn't go into here. I want to give, there are many collaborators, many of us around the country that have worked on FMT. I want to give a shout out to Mary Beth Nicholson and Stacy Kahn. And I started with a patient study. I'm going to end, a patient story. I'm going to end with a very quick story. So this is the nice thing about pediatrics. Your patients bring you drawings, and this was an 11-year-old boy who had recurrent C. diff infection, was impacting his life greatly. And on the day of his FMT, he brought me this drawing. He said, "Dr. Oliva, here is the C. diff the C. diff superhero. Here's the stool superhero who is actually slaying the C. diff monster. I want to make sure this works today." And thankfully, it did work for him. And it's just a reminder for those of us who treat patients that, we do what we do because we want to get our patients to better health and give them a better quality of life. So thank you very much for your attention.
Thank you so much for this outstanding talk. Are there any questions in the room? Dr. Goyal?
Thank you so much, Maria. This was awesome. I have two questions for you. With the new guidelines, what is your practice on use of steroids when the patient with acute colitis and positive C. diff has not quite responded well to vancomycin? And secondly, what is your practice in doing an FMT if a patient with IBD is flaring and has had suboptimal response to vancomycin as you escalate therapy?
Yeah, so, and Alka, I know that you are very involved in these issues as well, but this is what I would say, that for the patients that are flaring with and have C. diff, that we're following the adult algorithms, and steroids are not off the table, especially for patients with ulcerative colitis, that if we see that they are not getting significantly better over a matter of a few days, we're not afraid anymore to bring on the steroids, because those are the medications that, as you know, are probably going to be working the most quickly in patients. And we've now treated many a patient with C. diff, IBD, and steroids, and have noted, the improvements. And then I think your second question is, when do we pull the ripcord and do FMTs in patients with inflammatory bowel disease and C. diff infection? We're likely going a little bit sooner now, and we're not necessarily waiting for the third and fourth recurrence. The guidelines, as you well know, for using FMT without an IND and without a research protocol to be able to use it clinically, you still need to have recurrent C. diff infection, but we've moved things up a little bit more, and we're now doing it very often at the second recurrence, whereas before we would do it at the third and the fourth.
Any other questions?
Very exciting work. Do you see any differential between the ulcerative colitis patients and the celiac patients? You're lumping them, but I wonder, are there differences in response?
So do you mean the ulcerative colitis and Crohn's disease?
In Crohn's
disease, yes, patients. So interestingly, when it comes to C. diff infection, in the adult world, they noticed that the patients that were more likely to have C. diff infection were patients with ulcerative colitis. We haven't seen the same thing in pediatrics, and one of the reasons is that in pediatrics, actually, you have more Crohn's disease than you do ulcerative colitis, so that might be the reason. In general, colonic disease appears to be kind of the type of IBD that is more associated with C. diff infection. In terms of a response to fecal transplant, since most of the patients getting C. diff infection have colonic disease, and those are the patients that are getting FMTs, we really haven't seen much of a difference between Crohn's and ulcerative colitis responses. Yeah.
Thank you, Maria. What's your assessment of the safety of FMT in our more severe UC patients with positive C. diff testing?
Yeah, so at the end of the day, when we're looking at FMT, safety has always been kind of a very important caveat, something that we've paid attention to. What we can say, Michael, is that at least when we looked at all of our patients with inflammatory bowel disease in the larger studies, that there does not seem to be, first of all, the adverse effects after FMT are very low in pediatric patients, and even in patients that have the more severe disease. And we've also actually seen a very low rate of adverse effects in patients that are oncology patients, which may be even more severe than many of our IBD patients who've undergone FMT. So in terms of acute safety concerns, although we keep them always in the back of our mind, they haven't, raised the ugly head. I think one of the things that we don't know about fecal transplant is what is the long-term future. And I know, as you know, even when we do FMT for C. diff infection, we still need to tell patients on the consent form that we're putting them at risk for short-term or long-term conditions, maybe even immune-mediated conditions that they might acquire by us transferring the fecal microbe of the donor into the recipient. And so we don't know about the long term, but certainly in the shorter term, in a matter of months to a year or more, FMT appears to be quite safe in our patients with IBD.
Maybe one last question.
That was a fantastic talk, Maria. Thank you. I was kind of curious about one of your slides. I think it was midway through the presentation where you were showing the rates of C. diff in both IBD and non-IBD patients over time. And I noticed there was a bit of a sinusoidal pattern. And I was wondering if you guys were able to identify environmental factors or something else that had led to very high rates and then massive drops in rates and then back up. And it was a curious graph.
Yeah, no, it is curious, and you were paying attention, so you noted that. So that was more an issue of the numbers in terms of how the diagnosis of C. diff was being made, because it was, again, it was a database of discharge ICD-9 codes, and so we realized that that was probably impacting how the curves were just a little bit erratic, but in general, we were looking at the trends, and the trends remained that they were upward.
Thank you so much,
Okay,
Dr.
thank you.
Altenheimer. I have the pleasure to introduce another incredible speaker, Dr. Michael Snyder. He is the chair of the Department of Genetics and the director of the Center for Genomics and Personalized Medicine here at Stanford University. Dr. Snyder is a distinguished scientist and leader in genomics, renowned for his groundbreaking research in molecular biology. His research focuses on the complexities of the human genome and regulatory networks with significant contributions to understanding transcriptomes and transcription factor binding sites. Dr. Snyder's exceptional contributions have earned him the prestigious awards, including the George Beadle Award and recognition as a member of the American Academy of Science. Today he will talk about transforming healthcare with deep data and remote monitoring. Please join me in welcoming Dr. Michael Snyder.
It's a pleasure to be here. We are very embedded in many aspects of trying to understand nutrition, actually the colon. We're building single-cell 3D maps these days, which I think relates nicely to Michael's work. But I thought the topic I'd talk about today was using deep data, mostly in the area of trying to understand nutrition and looking at individual differences amongst us and how we react to the foods we eat. So what motivates us, I guess, starting this at the outset, is we think the healthcare system's broken. It's very much focused on sick care rather than healthcare. I think we'd all agree with that. But even if you think about how we practice healthcare today, all the steps we do are actually fairly antiquated, which is to say that you will typically get in a car, always at an inconvenient time, to go to a doctor's office, which looks pretty much the same as looked the last 40 years. A few gizmos might differ. When you're there, they stick a needle in you and draw a large aliquot of blood. And from all that blood and the time you're there, they actually don't make that many measurements when you get right down to it, some of which are a bit questionable, I would even argue. And then from all those measurements, they'll treat you based on population averages, which we think sometimes has value, but in many cases, it's really the individual, what's going on with you personally, that counts. And so we've been trying to actually change all of these different steps. And so as I say, today I'll tell you a little bit about using deep data, and I will focus on the nutrition side of things. So your health, as I'm sure you know, it's influenced by many things. Your genome probably counts for about 16% of your health span, with a pretty large standard deviation if you look at the data out there. But other factors, and you heard about a lot today, nutrition, exercise. We know intuitively these all impact your health. But we're in a world where we can now quantify pretty much all of these things. Activity and exercise, pretty good, actually with wearables, and we'll talk about that briefly. Food is a biggie. That one's still pretty clunky to log, unfortunately. Stress can measure indirectly. We're doing a lot with environmental exposures. All these things impact IBD and Crohn's disease, et cetera. In fact, we have some projects on the area of Crohn's and environmental exposure. Even if you don't measure these directly, you can obviously measure their effects by doing deep data profiling, and that is a big theme in our lab. So a number of years ago, it's actually 13.5 on me. I'm one of the participants, and then there's 10.5 on this group of roughly 109 people that we've been tracking. We sequence their genome once, and then we make as many measurements as possible out of their blood and urine, and we do a lot with wearables these days over the last 10 years or so. That's on top of deep clinical testing, questionnaires, and some advanced tests like stress echocardiograms, glucose control measures, and such. So we do take very, very deep data on folks, and then we also profile them over time. We'll sample them every 3 months while they're healthy, so it's really meant to focus on health. And then we'll take additional samplings at adverse events like viral infections, other sorts of things like that. And you'll see we run people through all kinds of fun projects. They're a really eager beaver bunch of folks who like to understand themselves, and we certainly try to help them do that. So anyway, as I say, we're doing all this. And one aspect that you might say, why do we do this? Well, we're trying to understand what it means to be healthy. How's it change over time? What's the difference between you and the person sitting next to you? How similar, how different are you? What happens at earliest times of infection? From the health standpoint, can you use these advanced technologies to better manage people's health? And I can tell you when we started this, pretty much every physician I talked to said, that's a bad idea to be sequencing healthy people's genomes. You're going to turn everybody into hypochondriacs. It's going to cost millions of dollars, et cetera. Well, we've warmed up from that somewhat. It's still a mixed camp out there on this. But I think it's a little more acceptable now. I tell you on wearables now, most physicians hate wearables because they think the same problem. Going to turn everybody into hypochondriac. We'll change that too at some point. Anyway, nonetheless, from all this deep data profiling, we did show, we were very curious as to its impact. And it turns out just from the first 3.5 years, nearly half the people learned something pretty important about their health. And no one aspect, I should say, it spanned a wide range of areas. You can see hem-oncology. Let's see, I don't know if there's laser pointer. Hem-oncology, cardiovascular disease, metabolic disease, and so forth. And all these were found pre-symptomatically. So in the case of hem-oncology, we found some with early lymphoma, 2 people with pre-cancers, 2 people with serious heart issues under cardiovascular disease. One was picked up from genome sequencing, another from wearables. And so that's another lesson. No one technology found all this. The idea was that a deep profile gave a much clearer picture of your person's health. And the idea, oops, I just took that slide out, is that if your health is a 1,000-piece jigsaw puzzle, we're trying to collect 500 or 600 pieces to get a clearer picture versus 5 or 6 we would argue you're collecting today. So we think you get very incomplete pictures. So the other thing that we can discover is that we can actually see how people age. And it turns out everybody ages differently because we're tracking them over time. There's about 600 markers that will shift with time, which we interpret as aging. And different people, as I say, age differently. Person number 2 there, their cardio age or their cardiovascular disease pathways, this one specifically, cardiac hypertrophic signaling pathway, shifts over time. The one on the left, they're actually typical age, their immune system, their metabolic systems all shift, which is pretty common. And we classify people into what we call ageotypes, where you can actually see their aging patterns. And we've now spun off a company that actually is part of it, what they're doing metabolic profiling, but they are tracking people's age. And again, same theme, you can see how people are aging. There are clinical markers associated with these ageotypes, so we think it is actionable. We actually have people who shift their behavior pattern based on this to see if they can modify their ageotype. We'll see how useful that is. Anyway, we're also very interested, so this is all nice, but we're very interested in this idea of how does lifestyle impact people's health, and can we modify their lifestyle to change this? And so our group, as I say, they're eager beavers. We profile them as they run through their VO2 max. We have a study like that out there to look at the effects of exercise. I like to say the things we do the most, we understand the least, like exercise, food. You hear a lot here. Sleep. We really don't know how those things work at a molecular level, if you want my opinion. So we're trying to dissect that. So I'm going to tell you some of the food work that we're doing now. So one area that interests a lot, which I think you've heard already, is this area about fiber being good for immune health, specifically feeding your microbiome and such. So we hear this a lot, right? And these are just some of the things this crowd knows this already. Fiber is very important for you, prevents all these diseases I've listed at the bottom. Feeds as I say, your microbiome, keeps it healthy, so you have good mucosal layers. But if you think about it, fiber is very heterogeneous. There's long chains, short chains, hydrophobic, hydrophilic, positive, negative. It's like saying all animals are the same. Well, at some level that may be true, but at another level they're very, very different. An alligator is pretty different from a dog, for example. And I think understanding the effects of these is pretty big deal. So we decided we're taking this from a very simplistic fashion to just start looking at two very common fibers. You heard about one already today, inulin. This other one, arabinoxylan. These are very common, arabinoxylan. I'm sure you have some every day, probably both of them every day. This is Symposium husk and other things, inulins in chickpeas and so on. And so we wanted to just see if we could see the effects of these. So we ran a fairly simple study, although it's actually a very deep study, so it takes a lot of time and a lot of resources. But we had 18 people who basically tested these different fibers. They were randomized of being either put on arabinoxylan or inulin, or then went on increasing doses, 10 grams, 20, 30 for 1 week each. That's 10 a day, 20 a day, 30 a day. And then they did a washout, switched over the other fiber, did another washout, went on a mixture. And so we then basically profiled them extremely deeply, the sorts of things we do. I was just telling you about with all the omics, looked at their microbiome and did deep clinical testing as well. And so what we were looking for is to see what happens when people eat these very, very common fibers. And so what you find is not a surprise. Thousands of markers change. Some go up, some go down. This is the effect of arabinoxylan. These are things that are generally following a dose-dependent response. So on the left are things that go up. On the right are things that go down. And then we can look at the pathways that change with these. Now, I forgot to say, the reason we picked these two fibers initially is that if you look at the literature, you'll find reports that say arabinoxylan is great for your cholesterol and it's great for your glucose. And you'll find other reports that say, well, that's not so true. Same is true for inulin. So the literature out there is fairly mixed. And what we discovered is that for arabinoxylan, in general, it definitely does decrease cholesterol. I think there is a consensus for that already. And in our case, we see a nice dose-dependent response. And the mixture also shows the same decrease. Inulin, in spite of the literature, some reports, had no effect overall. It turns out for glucose, neither actually improved the group overall. So I won't show that data. They're basically pretty flat. So that was not, we were expecting inulin might do that, but it did not for our group. We went on to look at this in a lot of detail, and I won't get into this too much, but basically we could show that the bioacids shifted. In the case of arabinoxylan, we could show that bioacids shifted in a way that ultimately led to a new model for how we think cholesterol works. I'll show you that in a minute. Each of these will actually shift the microbiome in these folks in very specific fashion. So this is inulin in the y-axis. That's arabinoxylan, if you will, with the, how much it shifts at the 30-gram doses. And the colored are the strains, if you will, that shift in response to one or the other. So the green ones are microbes that actually shift in response to both, so Bacteroides. And then the ones that go up, they shift specifically with inulin, and the ones that go that direction on the x-axis, they shift to arabinoxylan. So we can see what's shifting to what. And so we think that's actually useful when you're thinking about manipulating people's microbiome. From all the data we collected, and I mentioned the bioacid result already, we actually came up with a new model for how we think cholesterol is removed from you by arabinoxylan. We actually think it's degraded through microbiome and through this pathway that does involve your gut microbes. So I don't want to go overboard on that, but the bottom line is we actually learned a lot by doing these deep profiles. Okay. One of the coolest results, so comes back to this earlier theme, this population theme. Although the population as a whole went down with arabinoxylan, not everyone behaves the same. So this person on the left, they do go down with arabinoxylan, but the one on the right doesn't. Instead, that person's cholesterol goes down with inulin. So it's a very personal response. And we don't yet know, because our sample size is too small, what's driving these individual responses. We're now enlarging this. We're running 4 different fibers through people. You're welcome to join the study if you like. Does involve eating a lot of fiber, giving a lot of blood, but feel free to sign up. Okay, so we will see, and we're actually we decided we want to add a 5th fiber. So if you have a favorite fiber, you can throw it in there in the request list. I thought what was relevant to this was that, we run people through these studies and everything looks fine, but what tends to get ignored is actually 3 people that had a very, very strong inflammatory response to the higher dose of inulin. And that is in the literature, but it's not on everyone, okay? Again, there are 3 people that specifically reacted to this. I'm actually one of the 3. This is showing their liver enzymes went up, but if I showed you cytokines, one person, they just looked like a cytokine storm. It looked like they might have COVID if you thought. They didn't. It was before COVID. But the point out of all this is that you can actually see these very, very strong personal responses to individuals. And so that relates to the next sort of thing I want to tell you about, is that we've been working very hard to try and do what we call Amazonizing healthcare, if you will. And so many of you know we've been doing a lot with wearables, and I'm very keen on this. Thousands of devices out there. I'm down a smartwatch, I know today, because my Garmin broke, but I do have 3 others on, my Oura Ring. My hearing aid is for hearing, but it actually measures interactions and measures actually physiology as well. And of course, got to have glucose monitor and this sort of thing. So these are powerful devices because they follow you 24/7. And so I'll tell you about the glucose work. Again, I think that would interest this crowd. And the other area we've been pushing hard is this microsampling, the idea of taking pricks of blood, mailing it in, and measuring thousands of analytes. And I know what that sounds like, but ours does work. All right, so I'll just tell you one slide. We're doing a lot with the smartwatches for disease detection, especially infectious disease, and some of you may have heard me talk about this before. We think you can tell when you're getting COVID 3 days in advance of symptoms. We have a real-time alerting system that does this. But they do more than that because they're measuring 24/7, and they're fairly sophisticated devices if you think about it, right? These smartwatches are shining light on your blood, measuring absorption frequencies, and that's how you get SpO2. They're measuring a lot, and they measure things you don't measure in a doctor's office, like electrodermal activity, conductance on your skin, which actually has value for skin dryness. And if you're diabetic, I am diabetic, my skin is drier than average, and I can pick up signals. So these devices, they can pick up hematocrit, red blood cell count, hemoglobin levels. This is stuff we came up with through machine learning before it was popular. We could actually see all these clinical grade measurements from a smartwatch. I would argue they're not as accurate as clinical grade measurements, but they're good enough to detect these shifts from your baseline. So we think these are powerful. I think the most important monitor of all these devices is your glucose monitor. You may know 9% of people are diabetic and 33% of type 2 diabetic in the United States. I think the diabetes endemic is worse than the COVID pandemic. And so we think that these monitors could be very, very powerful for helping mitigate some of that. So a number of years ago, back when these were mostly just used for insulin-dependent type 1 and type 2 diabetics, we started putting them on normal people. How many of you worn a CGM? Really? 1? Okay, that surprises me. Well, maybe you all should wear one, I believe. Okay. We started putting these on normal people, and some people do have good glucose control, like the person on top. A lot of people are in that middle, where their glucose bounces around actually quite a bit. We call those moderate spikers. These, by the way, they're measuring your glucose every 5 minutes. They're just a little patch. I have one right here. They don't hurt. There are people at the bottom who are thought to be normal by hemoglobin A1C, fasting glucose, and they're spiking just as bad as diabetics. I think it's missed because of how they're actually measured. It's the form of diabetes they are. Type 2 diabetes is probably 50 different diseases. We can now subtype it, actually using CGM monitors of all things. Pretty cool. Anyway, we can classify people into glucotypes by whether they're low spikers, moderate, severe spikers. And then as did Aaron Siegel, went on to show that different people spike to different foods. So some people spike to potatoes, some to pasta, some to brown bread, some to white bread. This is from Aaron's work. The person up top that spikes to banana does not spike to a cookie. Probably the only person on the planet doesn't spike to a cookie. The one at the bottom spikes a cookie, but not a banana. That's more common. It's a very interesting selection of food that was sitting out there, I thought, today, some of which was good for you, most of which was bad. So anyway, we found something similar. Some people spike to bread and peanut butter, others protein bars, which have more than protein in them. If you don't remember anything from this talk, 80% of people spike to cornflakes and milk. That stuff is super evil. It probably should be banned. I think it's worse than smoking. Okay, it's not good. Okay, so anyway, I think it's really important. Again, it's very personal. Aaron's group has shown that 21% is due to your microbiome, but 79%, we don't know what it's due to. It's probably all those factors Alessia was talking about. This stuff gets all set early through your interactions with your immune system. So I think getting this calibrator right is a big deal and something we should be working on. All right. I can tell you, again, one reason these devices are powerful, if you wear one of these, this is a study, totally conflict, I forgot to tell you at the beginning, have all kinds of companies in this space. One of which is January AI, and they do have a CGM. And they showed just wearing a CGM for 10 days, people improve their time in range without even trying. Because you'll see, it's very eye-opening. You'll see what spikes you and what doesn't. So the obvious thing to do is eat the things that don't spike you, avoid the things that do. All right, here's the last story I wanted to tell you because I think it relates to this personalized nutrition. I really want to hammer this. And this is with this new technology that we've been setting up, we call home microsampling. And the idea is that basically you take these little pricks of blood. We actually spent 6.5 years working on this. We use two devices. One you get from your fingers are called neoterics. It's another from your shoulder called tasso devices. They both work pretty well. You basically collect very fixed volumes, fed exit to the lab, and then we do our deep profiles of lipids, metabolites, proteins, and we have targeted assays as well. We can measure about 2,200 analytes. The proteins and the metabolites with this kind of substrate are pretty stable. Lipids, it's a mix, but we know which ones are, which ones aren't. We can correct for those that are unstable. So then we went on to run fun studies, and this is where I think this crowd would like this. So we had 32 people drink this Ensure shake that you buy in CVS. Doesn't taste very good, I don't think. But anyway, we sampled people before, 30 minutes, 60, 120, and 240 minutes after drinking the shake. They were fasted before they drank the shake. Not surprisingly, hundreds of molecules change, but the cool result is we can follow some pretty important molecules. We can follow, these are glucose control molecules in cluster 1, insulin C-peptide, that's incretin, insulin, PP. These are all related to glucose metabolism. We can also follow inflammatory markers, interferon gamma, IL-4, leptin. Surprisingly, that one, in hindsight, I get it. Leptin went down. I didn't expect that, but now I get it. So anyway, we can follow all these things, including important molecules. So then what else did we discover? Well, it turns out everyone reacted differently to the same shake. So each box is a different person. This is just showing 3 carbohydrates. People tend to behave the same for the same class of molecules. So it would be nice to have the pointer. Is that a pointer? Yes. All right, I don't know if you can see that. So this person here, S13, their carbs plummet after they drink the shake. This person skyrockets, up, 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 flat, down, down. So different people are reacting differently, again, to the same shake. We can classify people into 5 groups. Gray is the average of all 32, and then each of these balls is a different class of markers. And the one that should interest this crowd is the inflammatory markers. A mixture of, we look at 80 cytokines and growth factors, as well as other inflammatory markers. For this group of people, the inflammatory markers go down. But for this group of people, they go up, up. So the same shake, pro-inflammatory in some, anti-inflammatory in others. And if you think about it, again, this crowd would appreciate more than most. 10% of people have inflammatory bowel syndrome. Most have no idea what's triggering it. We can actually measure that in a pretty simple and inexpensive assay. So I think we should actually be measuring people not just at fasting, but in response to all those things you're eating, at least commonly eating, because we could actually tell what's going on with you. Okay, this is what I view as the ultimate experiment. We have one person, you can guess who it was, wearing a CGM, a smartwatch, doing food logging, taking one sample with microsampling every waking hour for 7 straight days, 98 samples. And then what you can do is correlate all the biochemistry with the activity and physiology to see exactly what's going on, what's responding to what. You do these, we wrote algorithms to do what we call time lag correlations, because the idea is upstream events should be causative, or not always, but they can be causative of downstream events. But at least we can make those correlations. And not surprisingly, we find thousands of associations between, just even between heart rate, glucose, and steps. And these are, let's see, in magenta here is cytokines. These are lipids in blue and metabolites in green. It's kind of fuzzy there. But to list a few fun key ones, this is a bit of a sanity check. In response to glucose, your insulin C-peptide goes up, but we can quantify it. The magnitude, in this case, we can show it's 10 minutes after your glucose. That's when this goes up. It turns out inflammatory markers, that's known, go up in response to glucose, but this is a whole new one that hasn't really been described before. We can actually follow in detail what's going on with glucose-induced inflammation. And then here's a cool one, alpha-synuclein, which is implicated in dementia as well as Parkinson's. It also shows an interesting pattern. We think it correlates with certain kinds of stress. So if you are prone to dementia or Parkinson's, perhaps you'd want to know this and try and mitigate those effects, much like don't spike your glucose. This could be very useful. So bottom line is we're trying to actually understand personal responses so that you can actually have personalized behavior programs. And in fact, we are building personalized AI programs, as you might imagine. We have one for me. So you can see when you shift from your baseline and make predictions about, we can now predict glucose responses without a CGM from enough training data. Only 4 days of data will predict what your training is. And you can actually calibrate the whole thing with your smartwatch. So we think this tracking is going to be very, very powerful in the future. And to help do all this, we've actually built a dashboard. This is Amir Bahmani and others in the lab that pulls in your wearable, your omics data, your clinical data, all at a personal level so that you can actually follow it. So I'll wrap up. This is the wearables team and the microsampling team. That was when it was at its peak. It's not that big now. We blew it up when the COVID response came. Anyway, the point is I have an amazing lab that's actually been incredible for trying to implement some of the things I've been telling you about today. And I should have left enough time for questions if you guys have some.
Thank you for this amazing talk, Dr. Snyder. Dr. Kettleman?
Thanks so much. Super interesting. Have you learned anything either intentionally or inadvertently about intermittent fasting and either population level effects or individual level effects?
Yeah, we're running specific studies on that now. We have a general study that we run and analyze. It's not totally wrapped up around meal timing, activity, sleep with regards to insulin sensitivity and resistance and glucose spiking. And from that, there's a zillion lessons, some of which are obvious, but it really hammers it. Don't eat for 3 hours before you go to sleep or your glucose will be high the next day. Very common one. Take your walks after dinner for sure. Take your walks after you eat and you will lower your glucose the next day. Those are the kinds of things you can learn from these sorts of studies. For the fasting one, we're running ones where we're trying to do time-restricted eating for people. And not only that, we're trying to actually determine when they should do their time-restricted eating. Should you skip breakfast, or are you better off skipping dinner? The answer is probably dinner, but that's going to be the hardest to socially implement. It's a great question, though.
So, Michael, again, very inspirational, and you appreciate that.
Thank you.
Why I was looking for this. one of the major challenges that we're facing with personalized medicine is health for all. So in other words, equity. And I was wondering, beside the cost of implementing good diet is not affordable by a single mom with 2 kids at $10 a day, but coming to a wearable device that can monitor the adherence of things, I'm afraid that, we're leading to a Class A versus a Class B patients. What do you think that we, as a scientific and clinical community, should really do to mitigate this risk?
Yeah, it's a great question. I have several comments to that. One is I think the wearables are dirt cheap, and especially in the return for dollar. Imagine pushing someone's diabetes off for 5 years, which I'll bet you could easily do just by getting them to wear a CGM for 2 weeks. Again, I'm conflicted. We formed the company here. But anyone who's worn one of these will tell you it changes your behavior because you will see what spikes you. So I actually think, and these days they're $70, and that was this high markup. They'll be $5 in the future or less. So I think it can get out to the world. And on the wearables topic, you may or may not know, half the planet has a smartphone, even the poorest places. The reception may not be great, but they still get information. And so if you pair that with a smartwatch, you actually have a health monitoring system for half the planet. And these smartwatches don't have to be expensive. So I think it's an inexpensive way to bring health to the entire world. It's a way to democratize things. Now, the problem is, the second part of my answer is that the financial incentives the way we work now is totally misaligned, right, with health. So nobody pays to keep people healthy. We've got to change that because there's only 2 things that motivate people: money and family. So I think if we can you should get a discount if you wear a smartwatch. You should get a discount if you get your genome sequenced, I think, because you will better manage your health. And you can do economic models around cardiovascular disease and such where it really would pay to do this. Because again, $48,000 for 1 day in an ICU is not great. So I think we can save a lot of money if we just use a little bit of common sense, but nobody's paying to keep you healthy. We've got to change that. So finally, even Stanford now has a wellness program. They should give you a price break. They do give you a price break if you don't drive. So that helps if you take commute, but they should give you a price break if you actually wear a smartwatch. So I think that's the kind of thing we need, and I have other ideas in there. But yeah.
I believe there was a question in the middle somewhere.
Did you have a question?
Yeah, excellent, as always. You're measuring 1,000 parameters on one person. How reproducible is it across that patient? Have you done one person 10 times, 10 different weeks, and do you get the same patterns?
Yeah, they're fairly reproducible. The one we worried about a lot was the nutrition one in particular. If you grab the fasting, we're running a study where we have different people eating different carbs. Because of this issue, different people spike to different carbs. By the way, we all spike to white rice. There's another bad one. Yeah, and we run that in twice, and we see that the magnitude might shift a little bit, but not that much. Then we do mitigators. We're trying to see what will suppress spikes. What foods can you eat that will suppress your personal spike? That's also personal. We don't have all the rules worked out. That's something we're working on. And same thing, it's fairly reproducible. The mitigators as much as the spikers. So I think there are other factors, like I mentioned, exercising before a meal, believe it or not, will suppress your glucose spikes. So as long as you're fairly controlled leading into an activity, you can predict. But I think this is where we can do multivariate analyses, and this is where the continuous monitoring is so powerful, because it's easy to collect tens of thousands of measurements on a person over a week so that you can actually not only figure out individual activities, but multivariate, multi-factor activities and their responses. So I really think we can make proper models that are complex around individuals, because it's pretty easy to collect a lot of data very, very quickly around a single individual. That's where the personalized AI kicks in.
not eat their cornflakes with whole milk the second time around?
Yeah, well, they better take a run before or after they do not a run, a brisk walk, actually. Yeah, the one thing you may or may not know, strength training breaks down glycogen and you'll get a glucose spike. That's a good one, by the way. That's not a bad one. Yeah, so you have to pay attention. But soon you won't have to pay attention, right? Collect the data and let the AI tell you what's going on. Okay,
Thank you,
great.
Dr. Snyder.
All right. Thank you, Dr. Snyder. I think that really makes us think. Oh, I should stay away from that one. We should, how we could potentially change the way we design our not just our healthcare, but our own studies and research. I want to just take this moment to thank all the remarkable speakers we had today. I know it really got my mind going about potential collaborations, new ways that we might be able to make an impact on inflammatory bowel disease and celiac disease. I hope during the break and maybe during lunch that people are able to talk and think about how we can ignite new projects, new collaborations for these patients. So we do have boxed lunch outside. You're welcome to eat it outside, to bring it in here, to take it with you. Looking forward to connecting with some of you during lunch. For those of you who are interested, you can collect CME for this event, and there'll be an email about that coming to your inbox. There'll also be, this is our second event. We made a few changes from last year. We really appreciate your feedback, so you'll be a survey link in your email, and appreciate you taking just a few moments to let us know what we did well, what we could do better in the future, so we can make this as enriching and productive and stimulating a meeting for everyone each year. So thank you very much for joining us, and with that, we'll conclude.